like to welcome you all to the John Innes Centre. This is an event that started off aimed at getting some of the world-class scientists here to give talks about the amazing research that happens here, but in a way that was accessible to the staff that support that work, the people without a science background, without whom we wouldn't be able to do the amazing science that we do. And it soon became apparent, actually, that these talks would be brilliant to show off to science students studying at schools and colleges. And so we're very, very pleased to welcome in person this year. We've got T-level students from University Technical College Norwich and City College Norwich. And we've also got students from Wyndham College here, as well as the researchers and support staff from on-site at the Norwich Biosciences Institutes. But also we've got a hefty online audience, which Naomi is pointing in your direction at the moment. Last count, we had about 500 people registered to join us online from schools and colleges and an array of individuals from around the world, in fact. So welcome to you guys. Thank you for joining us. And do please join in. When you're online, if you want to ask questions of the speakers, please type those questions into the chat in the Zoom and we'll feed them through to the speakers. So this organisation was started by a group called the Research and Support Staff Voice at the John Innes Centre, and that's an organisation of people who work in a variety of roles who aren't necessarily scientists. We've been running since 2016 and the event's just grown every year, and it's great to see you all here. So for those of you who don't know the Norwich Biosciences Institutes, these are a group of research institutes all housed on the Norwich Research Park. The research park itself also includes the university and the hospitals and has an average daily footfall of about 30,000 people. The Norwich Biosciences Institutes in particular, though, are the John Innes Centre, where I work, which is a centre of excellence for working on plant and microbial sciences. The Quadram Institute next door, who do amazing science on human health and gut health and the gut microbiome. The Earlham Institute, who are a world-leading institute in genome sequencing and biodiversity, and our neighbours at the John Innes Centre, the Sainsbury Lab, who do incredible work on plant pathology and plant disease. So together we sit as part of the Norwich Research Park. And as I mentioned, we've got 3 local schools joining us in person, University Technical College Norwich, Wyndham College and City College Norwich, so thank you for making the journey. A little bit more detail about some of the institutes, or a look inside. Here's the Quadram Institute with their beautiful new building. We at the John Innes Centre are hopefully going to have a nice shiny new building like that soon. The Earlham Institute, which is right behind us here. The John Innes Centre, and here is our fabulous new building as it will look in just a few years' time. We're very excited about moving into there. And if we have a look at, we have the Sainsbury laboratory here as well, which will be part of the same beautiful new building. If you want to know more about the plans for the new building, the next generation infrastructure team have got a stand outside in the foyer, so have a talk to them at lunchtime. So today we've got a series of talks, and we've aimed for one long 25-minute talk from a researcher at each of our 4 institutes, and we also have a series of flash talks before and after lunch. These are given by staff from a variety of roles in a variety of career stages, just to give you a taste of some of the different roles here and some of the different career paths into science. And finally, we'll have our keynote speaker, Erica McAlister, who's kindly come from the Natural History Museum to talk about her deep love of flies. So, one note about the lunch break. If you haven't got a badge, there is no lunch. We will be watching. It's a 2-hour break, and that may seem excessive, but it's essentially because we have so many key support staff here that we need to make sure that they can actually do their jobs, as well as enjoying the talks. But for the second hour of the lunch session, for the students who've come from the 3 schools and colleges, we've got a Meet the Scientists session, which Shannon will be running in the Rec Centre Sports Hall next door. So thank you to all the scientists who volunteered to network with the students for that. I'm going to hand over to Kendall, who's going to chair the first session and introduce the speakers. So thank you, enjoy the day.
Good morning. morning all. We're not running too far behind time actually, I was a bit concerned, but if we do, we're going to allow our speakers their time allotted, around 25-minute long talks with 5 minutes for questions afterwards. I'll take questions from the audience first, and we've got lots of people online as well, so everyone online, please feel free to send some questions through. So yeah, welcome and good morning. I hope, like me, you are looking forward to our talks today. This morning's session, we have talks from speakers that represent a range of institutions and organisations across the research park. So you should come away with a good idea of the diversity of specialisms and career roles in both direct research and some science supporting roles as well. We start with the 3 longer talks for around 25 minutes with the questions that I mentioned already, and then following this we've got 3 flash talks from speakers who will be talking to us about pathways that have led them to the research park and their roles today. There's no question time left for them, but please do talk to them on the lunch and networking session if you have any questions. I'm sure they'll be happy to help to speak to you and share their experiences. Same goes for any of the speakers today or any of the scientists around. That's what this is about. It's about accessible science. So, in the interest of time, let's get started. To kick off our day, it's my pleasure to introduce Professor Anne Osbourn. Anne is a group leader at the John Innes Centre, whose research has greatly accelerated the discovery of new pathways and chemistries in plants. Anne was awarded the Novozymes 2023 Award in recognition of her scientific contributions, and beyond her impressive scientific achievements, she is also a poet and the founder of the Science, Art and Writing Initiative, which is a cross-curricular science education outreach programme. She also has published an award-winning poetry collection, Mock Orange, in 2020. So our talk this morning focuses on harnessing plant metabolic diversity to access natural products and their synthetic analogues, which can then be applied in medicine, agriculture and industry. Please join me in welcoming Anne Osbourn.
Thanks very much, Kendall, and welcome to the Science Park. So, I'm going to talk about something I've been working on for quite a few years now. And I'll get the hang of this. So, plants. You all know what plants are. They grow in the garden, they grow out there in the wild. And they're very, very talented. Plants can do incredible things. And we know that plants have been used as sources of traditional medicines for thousands of years. For example, Chinese traditional medicine. And so their medicinal purposes or uses have been explored for a very long time. This is a photograph of a chemistry lab in Germany in 1826, and it's the company called Merck. And I'm showing you this to make the point that advances in chemistry at that time began to make it possible to exploit the chemicals from plants and other organisms. And this was the foundation of the pharmaceutical industry, along with the chemical synthesis of compounds. So advances in chemistry were revolutionary in establishing the foundations of the pharmaceutical industry today. And having synthesized chemicals in the laboratory, the next step that the company Merck took was to develop a drug from a naturally occurring compound produced by poppies. And this is morphine. And morphine, of course, is very widely used as a painkiller. It's produced by poppies, and this picture shows you a photograph of a poppy field growing in Tasmania. And you can see that it's got a fence around it, and it's protected because this is a very high-value crop that is producing this compound morphine. So the global demand for morphine is enormous. So the next step then was to actually chemically synthesize a natural product in the laboratory. And this was first achieved for acetylsalicylic acid, which is aspirin. Aspirin is a relative of a simpler compound called salicylic acid, which is produced naturally by plants and is present in the bark of the willow tree. So the bark extract of the willow tree had been used for its health benefits, again, for thousands of years. So this is traditional use. But then chemists decided that they were going to try to make this compound, which they did, and this became the first chemically synthesized natural product-inspired molecule to be marketed as a pharmaceutical. But aspirin, on the left, is a relatively simple compound, whereas morphine, produced by poppy, is more complex. And so today, although aspirin is commercially synthesized, morphine is still accessed by extracting precursors from poppy plants growing in the field. So we're still not able to make morphine in the laboratory. So when we look at the kinds of compounds that plants can make, and don't worry if you're not a chemist, you can just look at these molecules and think, well, they look a bit complicated. They are complicated. Up at the top left, we've got a plant called wormwood, also known as artemisia, and that produces a compound called artemisinin. And artemisinin is very important because it's a component of treatments for malaria. On the right at the top, we have Taxol, which is extracted from the yew tree, and this is a potent anti-cancer drug. At the bottom left, we have another anti-cancer drug, in this case produced by a very pretty plant called mayapple or podophyllum. And you can see the structure. I don't know how to work the pointer, that's what I'm doing. You can see the structure of this molecule here. When we get to this compound, this is one that we've worked on for quite a few years, and it is horrifically complicated. And this molecule is very important because it turns out that if you put it into a vaccine, and you've all had vaccines, I'm sure, most of you will have done, it promotes the immune response. So you get a better response from a vaccine than you would if you didn't have this molecule in it. So it's part of a formulation for various vaccines, which I'll come back to later on. And this molecule is only produced by this tree over here called soapbark, which grows in a certain part of Chile. And these trees are not farmed. The molecule is extracted by taking the bark off the trees, and it's purified from the bark extract. So this is an example of a very, very important molecule that is only made by a tree that grows in a certain part of the world, and this tree is becoming endangered. So I showed you those examples to make the point that plants are really talented chemical engineers. They can make molecules that we can't make in the laboratory. And it turns out that over 50% of the drugs in current use are either natural products, meaning they're sourced from nature, from plants or perhaps from microbes, or they are natural product derived or inspired. So the plant kingdom, which is hugely diverse, produces a wealth of bioactive natural products. And there are over 400,000 species of higher plants on the planet, and that's a huge amount of diversity. And I've shown you examples of molecules that we know about. It's estimated that the plant kingdom is able to make millions of compounds, and most of them we haven't yet even discovered. So the vast majority of potentially useful molecules made by plants have not been discovered, and they certainly can't be accessed at scale. And trying to purify these compounds from plants is confounded by the fact that particular types of natural product tend to only be made in certain parts of the plant or under certain circumstances. So you can see at the top left violas with their pigmented petals, which are clearly partitioned. In the middle, we've got crocus, and the stamens are the source of saffron, which we use in cooking. And then over at the right, we've got the roots of ginseng, which produce these compounds, ginsenosides, which have health benefits. At the bottom, we've got two pictures taken under the microscope. The one on the left is actually a cross-section taken of an oat root, and you can see there is this bright blue fluorescent compound in the outer cell layer of the root. And this compound is antimicrobial, and it actually protects oats against attack by soil-borne pathogens, so it's a very important defense compound. And then on the right, we have a leaf of sorghum, and that leaf has been challenged by a spore of a fungal pathogen, and you can see that the leaf is responding by making these red pigmented molecules that are moving in vesicles towards the site of the fungal challenge. That APP means aprosorium, which is a fungal penetration structure. So there are two things here. These molecules are made in different parts of the plant or in response to different triggers. And also, plants don't just make these molecules for fun, because that would be a waste of energy. They make them because the molecules serve as attractants for pollinators, perhaps, or as deterrents for insects, or here, in protection against potential diseases. Another challenge is that if we grind a tissue of a plant up and try to extract a molecule, obviously we end up with a very complex mixture. And so here you can see a chromatogram on the right, where in this case a molecule of interest is a tiny little peak in the middle of a mess of other peaks. And so purifying compounds of interest can be very difficult when you start with a complex mixture. So given that we know that the plant kingdom is an enormous potential source of new drugs and therapies, the vast majority of which we haven't yet discovered, how do we go about understanding how plants make chemicals, the diversity of the chemicals, and how do we harness the chemical engineering capability of the plant kingdom to make the next generation of new drugs? Well, one advance is that with the, you've heard about the Earlham Institute, which does a lot of work on genomics. The number of plant genome sequences that are now available is growing rapidly. It's currently about 2,000. This slide is slightly out of date. These are the numbers of genome sequences for different plant species. And there are major new sequencing initiatives underway that aim to sequence the genomes of every species on the planet. So the plant genomes contain the DNA, the instruction manual, that contains the information that, if we could decode it, would enable us to make a whole swathe of known and unknown chemical diversity. So it's the instruction manual to make new drugs. So because of the limitations of chemical synthesis, I told you that even morphine still is not made chemically. How can we access this information from the plant genomes and then use that to make new drugs? And the answer, or at least one answer, which we think is very promising, is instead of using chemical synthesis, to use biosynthesis, to use the enzymes of plants to make new chemistry. Enzymes are very sophisticated, refined molecules that can catalyze the kinds of transformations that we simply can't do with chemistry. So what we need to do then is to find candidate genes for pathways that we think may be making molecules of interest, and then introduce these genes into a suitable host where we can put them to work and get them to make the chemistry. So one of the big advances recently has been in, firstly, the ability to generate and assemble high-quality plant genome sequences, but then you need to be able to scavenge through those plant genome sequences to identify the genes that you think might be involved in encoding enzymes that make chemistry. Your average plant has around 30,000 genes in the genome. Around 20% of those may be doing different kinds of chemistry. And we can use computational algorithms to scan through plant genomes to pick out the ones that we think are the top hits for doing interesting chemistry, and we can put those in a list of candidates. But that's all well and good. We're then stuck with a list of sequences of genes. So we need to be able to put those to work and get them to make chemistry. Traditionally, labs and companies around the world who work on natural products have used microbes such as yeast to try to make chemicals. But yeast is not a plant. It is a eukaryote, but it's not a plant, and it's missing many of the things that plants have. It's missing precursors and cofactors. It's missing the ability to precisely transcribe genes, translate them, localize proteins to the right parts of the cell. So although a huge amount of effort has gone into engineering pathways in yeast and continues to, it's not an ideal system. So yeast is not a plant, that's obvious. So one of the technologies that has been developed here, which is proving to be really powerful, is a plant-based system for putting these genes to work. So a colleague of mine, George Lomonosov at the John Innes Institute, was working on this plant at the left, which is a big leafy green plant we grow in the glasshouse. It's a wild relative of tobacco. It's called Nicotiana benthamiana, and it's very amenable to a process that we call transient plant expression. For transient plant expression, we take the genes from our hit list that we've got from our computational searches, We put them into plasmids, which enable the genes to be expressed. We put those plasmids into a little bug called Agrobacterium, which you can see at the top. Agrobacterium is simply a vehicle that will take those genes into the leaves of the plant, where it will then enable them to go into the plant cell and be expressed. So we're just using it as a little workhorse. And so we squirt the agrobacterium cells containing our genes into the leaves, and it's very quick. 5 days later, we can see what we've made, and here we can see fluorescent molecules and at the bottom, green fluorescent protein. And we can grind up the leaves and analyze the extracts and look at the metabolites. So it's very, very quick. It's called transient plant expression. It's not the generation of stable transgenic lines that we put out in the field. This is just a very rapid process that we do in the greenhouse. We can scale it up, so I showed you using a syringe without a needle to get the bugs into the leaves, but we can also use a vacuum infiltrator, and we can infiltrate whole plants. And over here we have Michael Stevenson, who was a chemist in my lab. He now has his own position at the University of East Anglia, and he managed to infiltrate several hundred plants and purify nearly a gram of 98% pure natural product. And this is his proud face, not his happy face, because it was a lot of work. Nevertheless, that's a major achievement. So we can go very quickly from putting the genes we want into these leaves and extracting the compounds. We can mix and match genes from across the plant kingdom, and we can make molecules that haven't been reported before. And using the exquisite capability of the enzymes in our toolkit, we can make selective modifications to complex scaffolds such as this one. And then we can purify the compounds from the leaves, and we can test their bioactivity in different assays. And here we have an assay for anti-inflammatory activity. So we've generated variants of that molecule, and you can see that they're behaving differently in these assays. So this is just a proof of concept to show that we can scavenge through plant genomes, pick candidate genes, make molecules, and test them. And we can start to understand the relationship between the structure of these molecules and their biological activity. There are even bigger scale-up facilities. If you go onto YouTube and Google, I think it's still there, there's a video, Medicargo vaccine. Medicargo is a company that no longer exists, not because of the technology it was using, but for other reasons. But it had glass houses in North Carolina in the US, where it had 40,000 plants on conveyor belts, going through a process of being dipped into this suspension of bug, infiltrated, and then afterwards the leaves were harvested. They weren't using this system to make small molecules, they were using it to make vaccines. Before COVID, they showed that they could go from the nucleic acid sequence of the flu virus to over 10 million doses of vaccine in under a month, which is very quick. And that was sufficient to meet the criteria for the US Defense Authority's rapid response to emerging diseases. Obviously, with COVID, everything's been accelerated to a whole other level. But the point is, this process is scalable. We can do it in the lab, we can do it with whole plants in the greenhouse, and there are these large-scale facilities, not just the Medicargo one, but others around the world. So if you come back to my friend, the tree that grows in Chile, this is the tree. It grows in central Chile, and it has very beautiful flowers, and the bark is the source of the molecule that I mentioned earlier on in the talk. So this is where it grows in Chile. It's quite restricted. You might remember there were a lot of fires in Chile fairly recently. There are dangers of this tree being destroyed by fires, by diseases. So there are concerns about the sustainable supply of this very valuable molecule that it makes. It does actually grow outside of Chile, and here it's growing in my garden, just outside Norwich, and it's flowering. And it has got seed pods now, but I don't know whether they're going to ripen, so I'm watching it on a daily basis. So the reason why this tree became so important for vaccines is a bit random. So this gentleman, who was a French vet in 1925, was trying to work out how to make vaccines perform better. And for some reason at that time, the feeling was you needed, when you introduced the vaccine, this is into animals, you needed to make this a more of an insult. And so he was trying things like breadcrumbs and tapioca and soy starch and alum. And he must have had a bottle of this bark extract, because the bark extract has actually been used historically as a source of soap and shampoo, which is why it's called the soapbark tree. So the West in the 1800s, I think, was very interested in having access to this stuff purely for washing purposes. So I haven't worked out how that bottle came to be in his lab, but clearly he threw it in there, tested it, and he got a really good immune response in his animal assays. Many years on, the molecule responsible for this has been purified, and this is the one I showed you earlier. It's called QS21. It's called QS21 because it was in fraction 21 of a fractionation. There's nothing magic about that. It was approved for use in human vaccines in 2017. It's currently in the Shingrix vaccine for shingles. I don't know if any of you have had that one. It's also in a malaria vaccine, a COVID vaccine, and another acute respiratory virus vaccine, with around 40 other vaccines under development. The current value of this molecule is between $100,000 and $400,000 a gram. So it's getting to be more and more important, and supplies are limited. So we wanted to understand how the tree made the molecule. And this is James Reed in my lab, who started this work. This was an emotional day when the tree arrived. And we sourced the tree from a nursery in Cornwall. It had been growing in the UK for a very long time. We didn't go to Chile and take trees. We weren't biopirates. So we had a tree sourced responsibly with all the paperwork to support that. We sequenced the genome of the tree with the Earlham Institute and assembled it, and you can see on the right-hand side cytology showing the chromosomes. And then we used a combination of approaches to scavenge through the 30,000 or so genes of the tree to work out which ones looked like they might be involved in making this molecule. And there were lots of computational tricks that we used, and a lot of knowledge of biochemistry. And as I said, the whole process of testing gene function is very quick using this transient plant expression system. So we could very rapidly test individual genes and combinations of genes, trying to work out the way to make this molecule. And eventually we did this. We were very, very pleased. It's a 20-step pathway, and we were able to take 20 genes from soapbark and put them into our wild tobacco relative and make this molecule QS21. Only at very low levels so far, but this is the first time it's been made free from tree, as we say. And so a lot of work is needed to optimize the levels of this molecule, but nevertheless, it's an exciting development. Interestingly, the tree makes over 100 structurally related molecules as well as QS21, including QS7 up the top there, which we've also made. QS7 is simpler than QS21. It's a very effective immunostimulant, and it's less toxic to human cells than QS21. QS21 has to be carefully formulated in vaccines. But the reason why QS7 is not used as extensively is because it's present at very low levels in the bark extract. But now we have the instruction manual from the tree. We have the potential to recapitulate the whole pathway for all 100 molecules, and also to make variants of them, with the ultimate aim of making next-generation vaccine adjuvants. So we work on a diverse range of plant species, not just the soapbark tree, and you can see some examples here. We've worked on understanding how wheat responds to attack by pathogens, and this induces a lot of defense-related compounds. We've worked on exotic plants such as pink shepherd's purse. Soapwort actually grows in the UK, and very interestingly, makes a molecule that's quite a long way in the direction of QS21, even though soapwort is not closely related to soapbark. And so we've also worked on that. We've worked on the tree on the right, which is a relative of the neem tree, which produces environmentally friendly anti-insect compounds. And so we're interested in how those molecules are made, because this could help to fuel the generation of environmentally friendly insect control agents. We've done a lot of work on Arabidopsis, which is the model workhorse that many people who work on plants study. And even though Arabidopsis is what you might call the gold standard plant that we think we know everything about, we discovered it made a whole array of 50 compounds, that it produces in the roots that influence the shape of the microbial communities around the root. So Arabidopsis is using these molecules to say, this is me, I'm a Arabidopsis, come and help me grow, or go away, I don't want you. And that's a whole other interesting area. And we've even gone into animals. Here you can see sea cucumber, which is one of the few animals that makes defense compounds. When it's attacked, it spits them out of its bottom to deter its predators, but which did not stop me eating a bowl of sea cucumber soup in China. So very briefly, I've talked about individual species, but now we've got a whole ton of information. We can use big data approaches, we can use artificial intelligence, we can use machine learning. There's a huge amount of computational work here. That is just, the opportunities are growing and growing. The question is, how do we handle all of this data? But there are smart ways of beginning to make inroads there. And just in the lab with different projects, we've made a whole suite of compounds, some of which we have in sufficient quantity to put into assays, which we've done, for example, with human cell lines, and you can see here just different biological activities. So I want to conclude then just by saying that I hope I've inspired you to see the enormous potential of genomics, computational approaches, the chemical engineering capability of the plant kingdom, to ultimately make the next generation of drugs and therapeutics. And we've set up a spin-out company, Hothouse Therapeutics, founded on this technology. And I'd just like to thank the many people involved and the sponsors. Thank you.
Amazing, Anne. That was really good and really inspirational as well, as you said. I particularly enjoyed the sea cucumber, strange-looking things. It's humbling to think as well, like how reliant we are on plants and the natural products that are out there. It's amazing to think what else is still out there. And the morphine, I didn't know we still got that from the precursor from the poppy still. Any questions from the audience first?
Did anybody spot a hydroxyl on a chemical structure that was in the wrong place? Feel free to say so. I'm not a chemist, I should say.
Penny.
Thanks, Anne. Fascinating talk. I'm just going back to your, so the Medicargo example. So during COVID, they were producing COVID-19 vaccines in tobacco plants. And many people might not realise that vaccines, we tend to raise vaccines in chicken eggs. So you'd think we would be primed for a plant-based alternative to that. And I remember having a conversation with someone at Medicargo, and he said, governments buy vaccines. And so do you think as scientists there's more that we can do to try and influence policy, to try and have this sort of push for more ethical approaches to producing these things?
Yeah, I think that's a very good point. I mean, there's the practical aspect of whether a vaccine made in a plant has the same properties as one made in other ways, and there are differences in the carbohydrate modifications, for example. The reason Medicargo went into liquidation, I think, was because it had a link with a tobacco company that the WHO didn't approve of, is my view. There's a lot of structures out there that are fixed in their ways. For example, the chemical industry does not like natural products because it frightens them. They're complex molecules, and medicinal chemists in big pharma look at them and think, well, what do I do with that? How do I access it? I can't work with it. And so, actually, the hit rate, if you use natural products to screen for drug activity, the hit rate is much higher than it is for chemicals that are made in the lab. But the tendency is to prefer the chemicals that are made in the lab because there is a feeling they can be controlled. But they're not encompassing the range of chemical space that is most fertile. So also, again, producing small molecules in plants is a bit of witchcraft because industry prefers yeast because it has big steel fermenters and knows how to grow yeast.
Yeah, it's fascinating considering you said it's not the ideal system to be doing that in. Any other questions from the auditorium?
We've got a question that's come from the online audience. This is from Rupert Koopman. As someone dialing in from a mega-diverse country, South Africa, how does your group intend assisting the persistence of potential medicine sources in the wild, i.e. the conservation of their habitat?
Yeah, that's a very good point. I have been to South Africa and been to workshops on natural products, and of course there is huge diversity of ecosystems out there and of chemistry. And there is a very important point here around biodiversity conservation. So the reason why I was telling you that we sourced the Chilean tree from a nursery in the south of England where it had been grown for 20 odd years with a clear paper trail is because there is a process called the Nagoya Protocol, which is very well intentioned, and its purpose is to ensure access and benefit sharing of natural resources and traditional knowledge. And so the UK signed up to the Nagoya Protocol in 2014, and that means if we access material from South Africa or somewhere else, there are procedures we should go through to work with local populations to make sure that there are benefits for those regions. And one of the things we learned from going there is that a really important point is that there is a big need for training. For example, in natural products, fractionation and in quality control, there are a lot of extracts of natural products that are used. They're very variable in their composition. So I don't know if that answered the question, but there's a huge amount to say about working with the plants of the world. We actually ran a science, art and writing workshop called The Global Garden, where we got people together to discuss examples of useful molecules that were originally produced by plants and of how they've been commercialized. And that was a very interesting exploration of case studies like the vanilla orchid, Madagascan periwinkle, and so on. Yeah.
Brilliant, thank you, Anne. Maybe time for one more quickly. Have you got any in the auditorium? If not, we'll move on. No? Okay, thank you once again. Thank you. So moving on to our next speaker, it's my pleasure to introduce Dr. Rowena Hill. She's a computational biologist specialising in the diversity and evolution of fungi, some really cool things. Rowena completed a PhD at Royal Botanical Gardens, Kew, where she exploited evolutionary genomics to explore fungal lifestyle and plant-fungal interactions. Currently, she's a postdoctoral researcher at the Earlham Institute, where she focuses on fungi associated with wheat and wild grasses. Today, her talk explores the important role of fungi in forests and also highlights the subsequent communication of science such as this in the media. Please join me in welcoming Rowena.
Thanks, Kendall. So hi, as she said, I'm Rowena. Let me get to my slides. I work at the Earlham Institute, which is just next door, on mostly fungal genomics. But actually today I'm going to talk a little bit about this debate that's really happening in the fungal research community and sort of what we can learn from it about how science is done and how critically it's communicated to the world. So just to make sure we're all on the same page, Anne gave us a bit of an intro there that yeast are not plants, but I'm just going to explain what fungi are. So this is a representation of the tree of life. And up there in the top left, you can see fungi. And so this is a whole distinct kingdom of life, and they're not the same as plants. So traditionally, it was thought that fungi were plants, probably just because of the way they grow out of the ground when we see mushrooms. But actually, fungi are more closely related to us animals. And just to give you an idea of scale, Anne mentioned that we think there are about 400,000 species of plants, and there are probably millions and millions of species of fungi out there. And they're absolutely everywhere all around us and interacting with everything. So microscopic fungi are inside and on the surface of plants. They're inside us animals, they're in our gut, they're living on our skin. The soil is full of fungi, waterways, fungal spores are in the air all the time that we're breathing in. Not many organisms can just survive living off rock, but there are rock-inhabiting fungi that can literally just live in rock. And there's even fungi frozen into the permafrost, so they might have been frozen in there for millennia and you can thaw it out and actually there'll be viable fungi that are still living and can be brought back to life. So they're pretty extraordinary, and they're actually also really useful for human society. So there's the obvious role they can play in that we can eat them. We can eat fungi, not just the edible mushrooms, but a lot of microfungi or yeast are involved in fermentation processes, so bread making, alcohol, cheese making, soy sauce, and then I'm sure many of you will have heard of corn, the meat substitute. So that's mycoprotein, that's actually made from fungus. And then there's all kinds of industry and manufacturing processes that are derived from fungi. So enzymes we get from fungi can be used in paper manufacturing, washing detergents, synthesizing biofuels, plastics even. Like one of the enzymes to make Lego bricks is actually from fungi. And of course, medicine. So Anne mentioned that yeast can be used as a tool to make medicines, but also a lot of antibiotics were first derived from fungi, immunosuppressant drugs, statins, and we're still early days of research into how to do it safely, but psychedelic compounds from psychedelic fungi even thought to maybe have potential to treat mental health illnesses. So fungi are really like the superheroes for our society, and I don't think they get the credit they deserve, but this is just sort of all the selfish stuff of what fungi do for us humans. Of course, they're also playing an amazing, important role just out in nature, in the world. And one of the things they do is they support our forests. So decomposes fungi, which we call saprotrophs, so sapro basically meaning rot, and troph meaning eat, so they eat rot, are just play an essential role in the world in that they can decay dead material. So basically we're not up to our necks in dead stuff. They can return all of the resources that are in that dead thing, back into the cycle of life. And this is actually not just so maybe when you go on a walk in the woods, you see, bracket fungi growing on a tree that otherwise seems to be alive. And that may actually well be a saprotroph, a decomposer. And it's actually just decomposing, it's not killing the tree itself, it's decomposing a bit of the tree that's already dead. So it's happening sort of at the scale of the canopy and not just on deadwood on the forest floor. So it's actually just a part of the whole cycle of a woodland. And also, you may have seen, when you go out and maybe you see one of those amazing ancient oaks that live for hundreds of years, they often have a cavity actually down the center. And this is caused by what we call heart rot fungi, so they rot the heart of the oak. And this is actually not necessarily a bad process. These trees still live for hundreds and hundreds of years. It's a normal part of their life cycle. And that cavity that's created by the fungi actually is basically a micro niche, a micro habitat that is then used by lots of other organisms like insects and other fungi and even plants. So so rot and decomposition is actually like an essential part of our forests and an important role. And it's so important that because in the UK especially, like our woodlands are so managed, often deadwood is removed, and we just don't have that many of these really ancient trees left. That this niche is actually kind of disappearing, is endangered, and some species are endangered because there's not enough decomposition potential. And so some scientists are even going out and inoculating trees with these heart rot fungi to encourage the existence of this niche. So that's the decomposers, but then I'm sure many of you will have heard that there's also another massive role that fungi play in woodlands, which is mycorrhizal fungi. So myco meaning fungus and rhiza meaning root. So these are fungi which have a close association with plant roots, and we call this association a symbiosis. And basically the plant and the fungus are trading goods. So the plant photosynthesizes, takes up carbon, and it can give some of that carbon to the fungus so the fungus can grow. And in return, the fungus can help the plant uptake water and nutrients. And so this is a mutually beneficial relationship. And actually, many of the sort of mushrooms that might come to mind when you think of fungi, things that we eat, or like chanterelle or boletes, and on the right, don't eat them, fly agaric, but they're very iconic, obviously. These are actually all mycorrhizal fungi. So they're obviously just the mushroom that we see above ground, but actually underground is the mycorrhizal fungus that has this symbiosis with plant roots. And this, this sounds like quite an amazing special interaction, which it is, but it's actually kind of the norm because over 80% of plant species have these mycorrhizal associations. So if anything, not having mycorrhizal association is the exception. And it's actually really amazing that so obviously they're playing an important role in an individual plant's health, but actually globally, they're probably underappreciated the role they're playing in basically just being in carbon sequestration, because we think of forests as these amazing carbon sinks. And actually this one study found that when looking at boreal forests, that actually the majority of the carbon that forest was sinking was because of the fungus, of the mycorrhizal fungus, and not the plant material itself. So mycorrhizal fungi, pretty amazing, pretty important to nature globally. But then there's also this is where we get to basically the debate that's happening in the fungal community, because some scientists have sort of gone this step further, where they say that they believe that this mycorrhizal network under the ground is actually able to connect many different trees across a whole forest, and that actually the trees are able to communicate with each other via the fungi. And when I say communicate, I mean chemically, you know, that the trees are able to send compounds via this network to each other, send resources. And it's given rise to this idea called sort of the mother tree, which is where a tree can opt to send resources to its offspring trees nearby via this fungus. And this has become like a very popularized idea. There's been popular science books about finding the mother tree, and this idea that, yeah, fungi can communicate with each other that trees can communicate with each other via these fungi. And it's also, this term has been coined, this very catchy phrase, the wood wide web. And that's been very widely reported, you know, in all sorts of media. And it's really like taken hold as an idea, I think, and like it's gone quite mainstream in the public consciousness. And it really sort of pitches woods as this cooperative system where things are all working together, which is very nice and romantic and idealistic, but is it true? And recently, different scientists have been pushing back against this idea. And so a review came out, a scientific paper came out a couple years ago, where basically they introduced some skepticism about this ideal system, and said that actually there isn't really the evidence out there that trees are able to communicate with each other in this way via fungi. And since then, another paper's come out that has many different scientific authors putting their name to it, and saying that, yeah, that maybe this is sort of a case where plants have been personified in this idealistic way, but is the evidence actually really there? And this is a quote from the first paper. They say, the claim that mature trees preferentially send resources and defense signals to offspring through mycorrhizal fungi has no peer-reviewed published evidence. So in that paper, they reviewed all the literature they could find, all the evidence and data they could find, where people studied the relationship between plants and mycorrhizal fungi, and they couldn't find any evidence that plants were able to communicate via the fungi in this way. A nice quote from the other one was, transfer from mother trees to seedlings would imply a remarkably collaborative fungal behavior with questionable adaptive benefits, especially to the fungus. So in short, what they're saying is, what's in it for the fungus? Why? So it's very energetically costly to do anything in nature. And the aim of any organism is just to survive. Everything wants to survive. Everything's is out for itself, really. Or that's our understanding. And so for a fungus to sort of, for no obvious benefit to itself, just sort of help plants shuttle resources across a wood, it doesn't really fit in with our understanding of how nature works, of how evolution works. And then, so there's this sort of other train of thought that's coming in a little bit more, which is that instead of it being sort of this beautiful cooperative thing going on, it's maybe more just a free market. It's like economics happening under the ground, where, you know, trees and fungi are in relationships and interacting, but they're out for themselves, and they're making trades depending on, you know, whether it's beneficial to them in that moment. And actually, there's some really cool research by some scientists where they are actually managing to image. So these are like the filaments of the mycorrhizal fungus, and what they can actually show is the stream of resources live that are going to the plant, and that actually in some moments they stop, and then maybe they go the other way. So basically what they think this is showing is that in real time, the fungus is, you know, changing whether it's giving its resources to the plant or not, depending on whether it's good for the plant in that moment. And presumably the same can also happen from the tree's side. And this, so just over the summer, there was this, like the major conference where all the scientists who are experts in mycorrhizal fungi get together and discuss all their research. And I really like this tweet that somebody posted about one of the talks, saying plants may do bet hedging with their roots. They may signal readiness to engage in mycorrhizal symbiosis in only one root at a time, while keeping the others non-symbiotic, just to keep options open. And I think that sort of idea, it fits in a lot better with our understanding of how nature works, how evolution works. The fact that, yeah, things aren't just going to do things out of the good of their heart, that they will be looking out for themselves. And cooperation can absolutely evolve. So, you know, mycorrhizal symbiosis between an individual tree and a fungus is absolutely, we have lots of evidence for this. This definitely exists. And it's because it's mutually beneficial, as I said, for both organisms. But this relationship between sort of different plants, that's where we don't really have the compelling evidence to show it yet. And that doesn't mean it doesn't exist. Sort of the point is that we need to generate that evidence. So this, in the first paper where they sort of critiqued this idea, they included this big box with a list of all the different experiments that could be done to interrogate this idea, to figure out what the roles actually are between plants via a mycorrhizal network. So, and this is, you know, this is how science works. This is how it's meant to work, that, you know, people have ideas and then we test them, we get evidence, and then we update the ideas. So it's not the issue is not that there might be an idea out there that is wrong. You know, it's good that the ideas are generated and then tested. The issue maybe a bit more is how this idea is being communicated. And how it's being communicated is not just on like the media or whatever, it's how scientists have communicated this idea to the world. So this is quite a nice article that on The Guardian, which I encourage you to read. It's sort of going over everything I'm talking about today. But I like this quote in it where she said, "The explosion of interest comes not from an unaccountable passion for fungal networks, but for what the theory implies, that the natural world is not static and cruel, but rather a living community governed by the same moral principles as our own." And I think that is one major part of why it's become so popular, this idea of the Wood Wide Web and the trees helping each other out. Because it's nice, we kind of want to believe it, don't we? It's kind of romantic, as I was saying. But it's maybe not that realistic, or at least certainly not nuanced enough. And the problem with how widespread this idea has become is that, you know, as humans, we have this thing called first impression bias or confirmatory bias, where we're often most likely to believe the first thing that we're told. And we can be quite resistant or defensive, even if we get new evidence and new information given to us that shows that maybe we were wrong and maybe we should update our beliefs. Usually we'll reject that. And that's like, we all do that. It's a very natural human response psychology to things. But the problem is, it will be a lot more work sort of adding the nuance back and correcting this idea to the public than it will be just having originally introduced it. And I mean, as I said, scientists perpetuated this idea. So this is just a blog. It's not even about mycorrhizal fungi. And, you know, it name drops the Wood Wide Web. So people, you know, out there were all using this Wood Wide Web because it is a catchy and easy to communicate message, and like somebody used this, and oh, it was me 4 years ago. I perpetuated this idea back. So we're all learning, we're all fallible people, you know, scientists are not perfect. And the important thing is that we are open to new evidence, and we update things, and we, wherever possible, correct errors that maybe we even have perpetuated. So at this point, maybe like, let's play devil's advocate. So I can imagine you might be sitting there thinking, well, why does it matter? You know, this idea spread really well and it got people invested in nature and fungi, and maybe it made them want to protect forests. If that, you know, increase in popularity and interest meant that more funding was given to the area and you know, more people got into it, maybe the ends justify the means. Does it really matter, that level of nuance? And I guess maybe it won't be surprising that as a scientist that I'm going to say this, but I would say that facts matter. And the reason that these sort of nuances in the facts matter is because governments use them to inform policy. And also corporations, you know, will ride on the back of the attention that the public creates from these sort of things. And I think a good example of where nuance is really important, or was really important in correcting policy, was recent tree planting initiatives where, you know, I feel like over the last 20 years, governments were saying, we're going to plant 1 billion trees because trees are really important for the environment. And huge initiatives were launched and happened where people were now just planted as many trees as they could without really thinking about, okay, well, what trees were they? Where were they? Were they being planted in already established important habitat that we actually shouldn't plant trees in? And actually now we know, and a lot of scientists had to work really hard to sort of introduce some nuance into that conversation to say that, well, yeah, it's not actually always the best to just plant trees no matter what, because for one thing, a lot of them might not survive if they're not planted correctly or in the right place. So then it's just a waste of money and time and effort, and it won't even work, it won't even be effective. And now I feel like there is more nuance, like governments understand a little more that, and we have this phrase you might see sometimes, right tree, right place, right reason, which is a much sort of, more informed view of like how we go about tree planting. And I think this sort of thing applies to the mycorrhizal fungal question too. You might have seen before, you know, it's quite big in the horticultural industry right now that people say, oh, well, you're planting this shrub in your garden, you can just throw a bunch of mycorrhizal fungus in that hole you've dug and it will help the plant do well. And that is, I'd say, almost never going to work or be the case. It's much more nuanced than that. And if we're going to, you know, protect our ecosystems, our forests, and do things right and not waste taxpayer money, then we need to make sure we understand the nuances of how mycorrhizal fungi are impacting forests, and we've got, you know, the whole picture. So what do I think we can learn from this sort of case study, this story? So scientists and science communicators, obviously we have the responsibility to get important results and important work out there into the world and communicate it well and clearly. But we also have to make sure that we don't sacrifice the truth by oversimplifying. I mean, it might be done on purpose, it might just be done accidentally, but we have to make sure that the results are not misconstrued, that we don't get sort of carried away with the oversimplified messaging, and make sure that, there's still enough nuance that things don't get misconstrued. Also, scientists are human, you know, like anybody, we have unconscious biases. We're not perfectly rational beings, and people make mistakes, and I'm not saying, you know, go away and never trust anything a scientist says again, but just that, everybody should exercise a bit of healthy skepticism or critique. You know, everybody should try and like fact check things that they read, no matter who is saying them. Also, all of us, we should all try and be open to changing our minds. Like I said before, this isn't actually that easy a thing. The natural sort of psychology encourages us to be quite defensive against this sort of thing, but actually it's very liberating, and it's a real strength to try and not let your ego get into it, and be able to, when you get new evidence that maybe means you should change your mind, just be able to see that clearly and be open to it. And finally, the Wood Wide Web idea is a lot more nuanced probably than has been communicated widely to the public and to other scientists. But I don't think it's any less interesting than the idea that's previously been communicated. This idea of like a free market, crazy economics going on under the ground between microbes and plants, the level of control they can exercise in whether or not they're trading with things, I think that's really fascinating and clearly more complex, but there's clearly so much more that we have to learn about it. So fungi are still really interesting, even if what you've heard about mycorrhizal fungi maybe isn't that true. So thanks so much for listening. If you would like to look at any of the slides here, you can get them there, and follow any of the links that I've included. And happy to take questions.
Thank you very much, Rowena. That was great to have an insight into the world of superhero fungi. And I do find it really interesting. I've always said, like, the big ideas you see in media are the most tangible ones to people and the public, and ones that run really quickly, and then it kind of snowballs a little bit. You just got to remember to step back and have some more critical thinking skills there. And it's not as though these interactions aren't interesting. They're just not quite as they've been portrayed. And it's easy to grab onto that really tangible idea, Any questions from the audience? We have plenty of time. Penny again? Yeah. I've got one on. Yep, sure. Okay. Yeah, you can go first.
So I've got a question online from Christina Hilken. She asks, if there is evidence for fungi and plant to form these important symbiotic relationships, but perhaps not between plants themselves, is there any evidence of fungi interacting with each other to find these resources and spots between roots?
So there's definitely, there's basically different groups of mycorrhizal fungi. There's ones called ectomycorrhizal fungi, which are on ecto, meaning the outside of the roots, and there's arbuscular mycorrhizal fungi, which form arbuscules, which is a specific structure on roots. And depending on the group of fungi, they can be more specific to a plant or more generalist. So arbuscular mycorrhizal fungi are very much more generalist. They can form association with this plant or maybe also with that plant and seem to be less specific. And different assemblages of these mycorrhizal fungi can form what we call common mycorrhizal networks. So there are interactions happening with where it's, it is like a soup under there. It's not like really clear cut. There are lots of different fungal species under the ground, and they're interacting in different ways with lots of different plants. So it is really complex. I don't know if there's direct evidence that fungi are interacting with each other, because presumably they're competitors in that landscape. So I don't know if there's evidence of direct interaction in that way, but there's they'll be interacting in some way. I don't know if it'll be peaceably, basically, yeah. Anyone else? Okay.
So my question was about, so first of all...
Oh, sorry.
First of all, fantastic talk. I think I'm one of your new fans. Fungicides, are they quite do they target specific fungus, or are they quite broad spectrum?
No, yeah, I think they're generally pretty broad spectrum. In the context of how does that hurt plant health if mycorrhizal fungi are good?
Yeah, so I was just wondering whether is there any relation to using fungicides and needing to use more fertilizers to get those nutrients in?
Okay, yeah, so basically, because fungi, there's this relationship that's been shown multiple times where using more fertilizers reduces the amount of mycorrhizal interactions with plants, because basically the plants are getting the nutrients they need from the fertilizer, and so there's no need to form that mycorrhizal association. So there's definitely a relationship there. In terms of fungicides, yeah, they do hurt the good fungi in the process of getting rid of the bad fungi. Although of course all these things are spectrums. Yeah.
Excellent, thank you. Any more online as well? No, brilliant. Thank you again, Rowena. Thanks. So next up for a final of the longer talks today, we've got Dr. James Canham, who has a very enviable job title of Entrepreneur in Residence. He's based at the Sainsbury's Laboratory, and he manages GetGenome, which is a non-for-profit organisation that supports researchers around the world with getting access to genomics technologies. During his doctoral studies, he developed a passion for collaboration and community, spearheading several projects focused on building scientific forums as well as outreach, which have all led him to his current role today with GetGenome. So please join me in welcoming James.
Thank you very much. Can you hear me okay? You can hear me okay? Yeah, great. Well, first of all, thank you very much for being here. It's amazing to see so many people here. I've been on the park for about 10 years now, and I've seen this event grow from very humble beginnings to what it is today. So real credit to the organising committee, both past and present. And thank you for the invite to speak. So yes, my name is James Canham. I'm an entrepreneur in residence at the Sainsbury's Laboratory. So I help to provide some support to some enterprises that are developing from the amazing research that happens on the research park. But I've had a very specific focus over the last several years, and that's with GetGenome. So GetGenome is our non-profit organisation that has developed from the labs in the Sainsbury's Laboratories, supported by TSL, but is also independent. I have a few links dispersed across the talk that could add a little bit more context to what I'm saying. So perhaps go and check out some of that information behind it, particularly the people who are watching online. So what is GetGenome? Well, as I mentioned, it's a non-profit organization, and our mission is quite simple. We want to empower researchers, empower scientists around the world. The way that we do that is we provide them with access to genomics technology and genomics training. So you heard from Anne the importance of genomics, so DNA sequencing at its core when you initiate projects and how much we can learn from DNA sequences. But unfortunately, a lot of the global research community doesn't have access to those technologies to acquire the DNA sequences of the organisms that they're interested in. So we exist to support them. What is genomics? Well, this is Manel. Manel's a Tunisian research scientist that we supported early on during the conception of GetGenome, and she's been a real amazing ambassador for the organization. And Manel says that if I want to identify my bacteria with whole genome sequences, I have the whole story. As a scientist in the field of biocontrol, knowing the system of interactions between my bacteria and the host is crucial. The genome is the key. So acquiring that DNA sequence is the real start point for these projects. We've been living in a genomics revolution. You're probably aware of it. And it was really catalyzed by the Human Genome Project, the first draft of the Human Genome being published in 2001. And this was a real technological revolution. And it drove this dramatic reduction in the cost of DNA sequencing over the subsequent 20 years since the first draft was published. So back in 2000, it would cost perhaps hundreds of millions to sequence a human genome, whereas today it might cost a few £100 and take a few hours to do. But it's only a revolution if you can experience the benefits of that. So, as I mentioned, large swathes of the research population do not have access to those technologies, and I think this was most keenly felt during the COVID-19 pandemic. Strains of concern could arise indiscriminately, so randomly across the globe, whether a research community is in a position to be able to identify it and monitor it or not. And this is reflected, I think, in this database of SARS-CoV-2 sequence data. This contains over 15 million sequences of SARS-CoV-2, and as you can see, the vast majority are from Europe and North America. Unfortunately, sequences derived from Africa only contribute less than 1% of this database. So there's a real problem here that we have to address. It's in our view that GetGenome is a part of that story to start to address this challenge. And we have a very simple workflow. So Anne mentioned about the Darwin Tree of Life and some other projects, and they're very much needed, and they're top-down approaches. So at government level and then institute level, what GetGenome is a bottom-up approach. We help the researcher first with the challenges that they're facing, we support them with the research that they're focused on. We have a simple workflow. It's deposit their organisms with us, and we help them access those DNA sequences, and we provide that data to them free of charge. We then engage with them through a series of workshops, training opportunities to help develop their skills in genomics. We then help them share that information with the community, because research is only part of a researcher's job. The other major part is disseminating that research more broadly. And then finally, we want to bring these researchers together. So what we commonly find is researchers that are disparately located around the world are often interested in very similar organisms for similar purposes, yet they don't really talk to each other. So we bring them together under one umbrella, under a network, and this gives them new learning opportunities and collaboration and partnership opportunities. Really, it's about the people. It's about the researchers, and I've been incredibly fortunate to meet some incredible, really, really talented researchers around the world working for GetGenome. There's another issue that I would like to highlight, and it's one that you might not be so familiar with. That is the cost of publishing research. It does cost a huge amount. It costs institutes a lot of money to publish scientific research in journals in a traditional way. And it also costs a lot of money for institutes to subscribe to these journals to access the data. So I think you probably understand where I'm going with this. If we've got under-resourced research communities out there, Often they can't publish their own work. Often they can't access the data that they need to progress their research. So that presents another real challenge. We tried to overcome that at GetGenome through primarily training, but we've also brought together a community. So first of all, we help them disseminate their data on open science platforms, of which there are plenty. And then we create a community where they can publish their work, their articles, which are sometimes the only scholarly output that they might produce from a project, onto this community for all to see, completely free of charge. So they can deposit and they can access without cost to them. So open science is incredibly important, I think, in research. And at GetGenome, we're keen proponents of this movement. So I want to tell you about a specific project. This is our work in Tunisia. So we launched what we call a call for projects. So this is the sort of model that we work with, typically on a campaign. So we create a scope of a call. So we ask the research community to apply to have support from us. So Tunisian researchers could apply and have their genome sequences created and then provided to them. So through this call, we brought together about 30 researchers in Tunisia. Their main focus was non-pathogenic bacteria, and we created over 130 genome sequences. We engaged them with a series of training workshops, and ultimately, what we're left with today is a whole lot of data that is now published on open science repositories and available for the entire community, and lots of articles that have been published from these scientists, which is a really fantastic output. You can see a picture there of Professor Sofiane Kamoun. Sofiane is a group leader at the Sainsbury Laboratory. He's also a founder and trustee of GetGenome, and he's with the Tunisian ambassador to the UK. So the broader community really did support GetGenome and recognized the impact that we were having on the ground. And this really gave us a model to then use in other parts of the world, and we have done that. So to date, we've supported around 100 researchers in over 20 countries around the world across a series of different campaigns that we've developed. I want to just briefly tell you about the type of researcher and research that we're supporting. We don't conduct any research ourselves directly. We only support those scientists around the world on their projects. But our call in Pakistan is quite typical in terms of demographics. Around 50 to 60% of those researchers that we support are traditional early career researchers, so PhD students, sometimes undergrad students, and postdocs. Around 40 to 50% are usually associate and assistant professors. And often those associate professors are typically creating their own lines of research, they're independent teams. And so by helping them, we're often helping a number of students and a larger group. The types of research that we have supported varies from place to place, but because we've typically focused on non-pathogenic bacteria, many of these projects are concerned with growth-promoting bacteria, for example. So these are microbes that are used as biocontrols in the field to help protect plants against biotic and abiotic stresses. So they're going to be an incredibly important source of bioprotectants, potentially pesticide alternatives for sustainable agriculture going forward. Other projects include bioremediation, so cleaning polluted soils and waters of harmful chemicals. So microbes can be used for that. Probiotics, so the healthy, the gut bacteria that are so important for human health. And lots of other projects such as bioproduction, bioengineering, and enzymology. So it's a broad range of different research projects. And we've also engaged with those researchers in a number of different ways. So we have helped to run lab practical sessions, in-person and virtual workshops, conferences, and even in field work as well as a part of one or two of those campaigns. So that is the first sort of major part of my talk. And first of all, I want to encourage you to head over to whichever social media you prefer and follow GetGenome. I would like to thank the contributors of GetGenome, those that have really helped us develop this organization into something that we're really proud of. Joe, Adeline, Pi, Mia, also Andy, Amal, and all the Commune Lab, and the wider TSL community. And our founding trustees as well, who have a huge amount of experience in research, in collaboration across the world, and working with Genomics, Saskia, Sophia, Nick, and Apollinaire, who brought GetGenome from a concept to a real entity that's helping researchers around the world. So finally, I actually put together my own little flash talk, because my journey to this point hasn't been very typical. And I use the graph of contentment as a way to describe my journey. And those that did a PhD here may be familiar with this concept. So this graph of contentment is my contentment over time. To be totally honest, I only ever think, in terms of success, I only ever think of contentment as being the major output, the key factor. So this is how I move through my adult life. But we start here, me as an unfocused 15, 16-year-old, a high schooler that was really desperate to leave school, in truth. I wasn't particularly focused, but I found my passion at that time in an unusual place, and that was actually on a golf course. So I used to cycle to my local golf course. I would put a strimmer on my back and go out and strim banks, grass banks and cut hedges and this sort of work. And I found that work incredibly enjoyable. I was out in nature. We had a fantastic team, and I was lucky. I had a manager who said to me that there's a career to be experienced here. And so inevitably, I found myself back in school, only a short time after declaring that I would never go back to school. And I studied vocational qualifications. And because I was passionate and I worked hard, I was noticed, and I actually got a job offer in Switzerland. And I moved over there, and after a brief spell of horrible homesickness, I barely left Norfolk, in truth. I started to find my feet, and who wouldn't, in the Swiss Alps. Absolutely amazing place to work. And I took over the team, and I was top of my game, thoroughly enjoying life. But there was something nagging at me, and that was that I didn't fully understand why my plants, why the turf was reacting in the way that it was to different stresses. Sometimes we would have a hot summer, and my grass would die, and the members would be shouting. And other times, we would have pathogens, and I would come in the morning dew, I would see these little hyphae glistening, and I would think, oh dear, this is going to be a problem, and in a few days, my greens are completely brown. So I needed to learn, and I knew the only way I could do that was to return to full-time education. And so I came back to Norfolk, to the UEA, and studied plant science, and it was probably the hardest thing I've ever done in my life. I had to learn how to learn again. But again, I worked through that, and I was very fortunate to have some amazing lecturers, amazing friends there, and 5-year course soon flew by, and I ended up completing a PhD at the John Innes Centre in a plant science-related field. And it was an amazing pleasure to work through that. During my PhD, I used genomics extensively, and so I understand the transformative power that genomics can have. But I also experienced 3 months in East Africa, where I learned about bringing groups together to try and tackle some major problems, and also utilizing genomics to help us with those challenges. So we were looking at a particular disease of coconut at the time, and I brought together groups both in the UK and in East Africa and we worked on this project. And I guess that was a precursor to the next steps, because I found myself at TSL, and yeah, it's a learning curve. You come out of education and you have to, again, you're thrown into an unfamiliar environment, and you've got to push through. But I have to say, seeing the impact of GetGenome, what it's done, what it's doing on the ground, and how it's supporting researchers, I wouldn't change anything. And my interactions with those that I meet are overwhelmingly positive. I tend to learn more from them than they learn from me, and that's wonderful. So the decisions you make are not deterministic. You can always change direction. You can always pursue a passion, and that's really important to do. So you don't always make the right decisions, but making a decision is living, isn't it? So thank you very much. I'm happy to take some questions.
Thank you, Joe, for such an engaging talk, and for being honest and sharing with us about your journey to where you are today. I think it's quite nice to hear when people have very non-typical pathways to where they are, and I think you're told a bit differently, sometimes, especially when you're a young person, that it's just one straight trajectory up, and I don't actually think in reality that's real for most people. So it was really nice to hear your story. Is there something else I wanted to say to you? I had a question. Sorry, anyway, let's take it to the audience for questions instead. Anyone? Over here?
Fantastic talk, thank you. And I love the contentment over time. And I guess contentment also correlates nicely with success as well. If you're happy, then you're often successful. So I was wondering what next? You've got a really cool job title, Entrepreneur in Residence. I was wondering what the plans are next.
Well, I'm very fortunate that I get to run an organization, but I also get to experience science and research, and I'm exposed to that world as well. And so I see the amazing work that's happening in the research labs. And I also see the demand, the huge demand to translate that work, that research, into commercial outputs, into other entities. And so I'm very excited to be an entrepreneur in residence. I guess I fell into that role, and that title perhaps doesn't fully describe what I do on a day-to-day, right? I run a non-profit. But yeah, I hope to be in a position where I can support those entities as they try to find their way, you know, in a challenging world. But ultimately, if I'm not supporting people, and I've learned this about myself over the years, if I'm not supporting people, I'm probably not content, and therefore, whatever I do in the future is going to be helping people somehow.
Thank you, James. Anyone else? In the middle, Neil.
Thanks for a great talk. You mentioned that in Switzerland you grew a bit dismayed by what was happening to your greens, and then went back to learn more about biology. Did you learn more about golfing greens and how to keep them green and lush?
That's really interesting, a really interesting question. You know, people ask, the moment you turn up at university, especially as a mature student, you're asked, well, what job are you going to do at the end of it? I didn't care what job I was going to do. I was there to learn. I'd given up so much. I'd given up a successful career in the Swiss Alps, you know. So it wasn't ever about a job, and it was about learning. One thing that I was very and I mentioned the lecturers. We had this place on our doorstep, and actually I didn't know much about the Norwich Research Park. And when I turned up to my first proper year, I had to do a foundation year because I had no A-levels. I learned that actually a lot of the research is about grasses, is about wheat, is about barley. So there's an awful lot of crossover of translational information there, for sports turf. So if you're perhaps a golfer, I can maybe hear it, I don't know. No, you're not. But yeah, there's an awful lot to learn. We learn, how plants interact with pathogens, how they interact with their environments is broadly the same, with nuances in various systems. And golf courses are very unnatural environments, and so those turf grasses are under a lot of stress, actually, a lot of the time. So that, yeah, I mean, there's a lot. I learned a lot about it. And maybe one day I'll return in a different guise.
Thank you.
One more over here as well.
Thanks. It's wonderful to hear a mature PhD student. I was a mature PhD student myself. We need to stick together. Yeah. You showed the global emergence of different SARS strains. And one of the things that really frightens me is the poor quality of disease surveillance in the global south and the network for that. So I would imagine that your work is contributing to that. How do you see the future with that?
Wow, yeah, it's a big question. There's no doubt that is a huge challenge we face. And the reason I used the COVID-19 pandemic, I know we'd like to forget about it in many ways, but it really did highlight that imbalance. And as you say, the lack of resource to monitor and surveil for these diseases. It should be said, for those of you that are unfamiliar with plant sciences, plants are experiencing epidemics, pandemics all the time, and there's some really, really major problems with our food security unless we can properly surveil and identify these pathogens and learn to understand the biology. We have a number of campaigns that will focus on specifically on monitoring, and actually we're working with a very large consortium on wheat disease, which is affecting parts of Southeast Asia and Central Africa. And again, though, I bring it back to building that capacity from the ground up. I do love those big consortia that are trying to filter, large amounts of funds and resources down from the top. But ultimately, if we're not studying those organisms that are actually causing the harm on the ground and are actually the research focus, then a lot of that resource is going to be wasted, I think. So that's why we're going for this bottom-up approach. I hope I've sort of partly answered your question.
Thank you.
Thank you.
Thank you. Any more questions from the room? No, we'll go to online then.
Thank you. James, we've got bear with me. Question from Taiwo. Thank you for this talk. Fermentation is an important aspect of processing plants to bioproducts. How can genomics transform this area of biotechnology?
Does somebody else want to? Where's the scientist?
I mean, I guess you mentioned you've been sequencing organisms that people were using in fermentation and biotech.
Okay, so fermentation. So I guess there are some research. I guess there are some researchers that we're supporting that are interested in fermentation, and of course, I mean, genomics. It's incredible how it really does or should act as a start point for many of these projects. We have to understand the foundation, from the genetic material right through to the phenotype. And so it's one area, I guess, that we will support indirectly, but I can't really answer it specifically particularly well. I think you can see I'm floundering. I'm a greenkeeper.
Well, we've got one. Well, in fact, we've got a nice little comment for your talk, which I thought I'd read out online. Not a question. This is from Helen Dorrell. Amazing career route, bringing all of your skill sets together through hard work. Inspiring talk. It's never too late or too old to return to formal study. Learning is lifelong. Thank you.
Thank you very much.
thank you. So we're moving on now to our flash talk section of this morning's session. We have 3 flash talks for you, generally on the topic of, again, kind of career trajectories and pathways, and maybe something that's a bit non-typical that you wouldn't necessarily think would lead people to where they are today. So first up, we have Mikhaela Neequaye. Yeah, got it right. Sorry, Mikhaela, I didn't have time to talk to you before the beginning. She is a research assistant at JIC. These talks will be about 5 minutes. We might have time for questions. We're running on time, a bit ahead of time. But the idea is you don't. So welcome, Mikhaela.
Right, thank you for the introduction. I'm Mikhaela Neequaye. I'm a research assistant here at the John Innes Centre, and I'm just going to speak to you briefly about what I do here. So this is my very canonical career path, which took a lot of turns, but essentially I did my bachelor's in genetics, and like a lot of people, kind of started my career as a biologist, not really thinking about plants. It wasn't until I got a job in the botanical gardens at the University of Leicester that I even realized that my love of genetics and studying molecular genetics could be married with plants. And so I went running headlong into a PhD without having done a master's, and was really fortunate to do a PhD here at the Norwich Research Park. So I was co-supervised by the Quadram Institute and also the John Innes Centre here. And then COVID happened and all sorts of things happened in between. And I've ended up as a postdoctoral research assistant here at the John Innes Centre. So a full-time researcher. And I want to tell you a little bit about my science because I love it. And it actually draws together all 3 talks somehow. I like to say that I study the chemistry of chemistry. And that is to say, the way that chemicals are used by plants for attraction, specifically for pollinators. So Anne was talking about the beautiful, rich diversity that there is in plant natural products, and that they're not produced all the time. And my focus is in those smells and colors and interesting chemicals that are made when it's time to reproduce. And that is the way that plants are always trying to get animals involved in their sexual reproduction process. I study floral scent and pigment, and what I've done is I've brought some floral scents along, so when we do the meet and greet at 1, you can come and smell some of those scents, including the orchid that I work on, which is here in the Swiss Alps, where James was, and also Saponaria, which Anne mentioned. So part of studying floral scent isn't very glamorous. Essentially, you just trap a flower in a chicken roasting bag, because it's very inert. Our lab must be the largest consumer of chicken roasting bags in the UK. And then we use a pump, and we suck the air through off the surface of the plant without doing any damage to the plant. We run that on what's known as a GC, gas chromatograph, and we produce a chromatogram, which is what Anne showed you as well. So my science is all about studying the evolution and the diversity of floral smells, which is fantastic. I look at pretty flowers all day, and I think about how pretty they smell. The other thing that I do here at the John Innes Centre is I think about diversity in a different way. So I'm the chair of the READ committee, and I'm really, really fortunate that the John Innes Centre is the kind of employer that really commits to promoting these initiatives. So that is locally, it's our Centre for Race and Ethnicity, Equity and Diversity, and I implore JIC staff and students that are interested to come and get involved, because this is a really wonderful place to try and get some work done in that field. In the wider NRP community, there is a Black Health Conference happening this month. It's Black History Month. Happy Black History Month, everyone. And that will feature speakers talking about Black health, maternity outcomes, mental health, and also research journeys. And Evelien from Bake Off will be there giving a talk. So if that doesn't sway you. But I'm also involved in national efforts to promote equity and diversity in science, and that is the Black in Plant Sciences Network. So this is a network of Black plant scientists from all across the UK, which you would think sounds massive, but there's actually only 2 Black plant science professors out of the 23,000 professors present in the UK at the moment, which is to say plenty of work to be done, and lots of people, including the JIC, including Black in Plant Sciences, doing that work, when I would go to talks, I wouldn't really see myself reflected in the science that I was seeing. And if there's anything I want to say to particularly the students here today, you might not see yourself in the people who are presenting here today, but that doesn't mean that you're not meant to be here, because you are. There are people doing the work to make sure that you will be welcome. And also, you know, we just can't wait to see what you all do. So thank you very much. I will be here for the meet and greet at 1 till 2, I think it is. I've got some smells, and I'm happy to talk about these initiatives as well as all the science that I'm doing and all the things that I did to get here. So thank you very much.
Excellent, Mikhaela. Thank you for a really nice flash talk. You're clearly very passionate about what you do, and it's really nice to hear, and hopefully you all enjoyed it too. So next up, our next flash talk is from Matt Castle. Matt is a horticulture specialist at the Sainsbury Laboratory. Please welcome Matt.
Good morning, everybody. I think the title of this talk is going to be that you cannot make a bad career choice. And I will try and explain now with this talk, hopefully not fail. So here I am at 3 months old. I'm just reacquainting myself with my twin sister. We've already met, forgive me. This isn't supposed to be on roll. I've just had heart surgery. This is going to come up again in my career, and it's going to be quite pivotal in my adult life. Here I am at 18. What's crucial here is, like many of you that are watching this presentation, I've got a Saturday job, and I'm stacking shelves at a local supermarket. And in that role, I will learn about these products, and I will go away to university and fail university doing a plant biology degree, ironically. And when I come back, I will apply for a role as a cosmetic chemist. And I will talk my way into that role because I was putting those products on a shelf in a supermarket. And in those products are raw materials that will play a pivotal role in my adult career. I hit 25, I become an adult. I never thought it was going to happen. And I go away to university and I study commercial horticulture. While studying, I have a job as an agronomist. If we look at some of these growing techniques here, they will come up again in this presentation, as will moths. This helped with the technical side of my degree. A gardening job helped with the practical side of my degree. And this is the castle I worked at in Kent. And a pub role helped with public speaking, but I'll let you guys be the judge of that. On completion of my degree, I became a plant health and seeds inspector. ISO accredited. We ended up becoming so good that we got to travel all around Europe, training with other inspectorates. Again, using some of the chemicals that I first experienced in cosmetics. All was going really well. I got promoted, and I also elected to have heart surgery for a second time. This is apple juice, okay? It's not beer. However, that made me re-evaluate my life. I wanted to go back into horticulture, so I became a hydroponics manager on the remote island of Ascension. Here we have the vine system that's not working very well, tomatoes. Here we have the leafy vegetable system that worked perfectly. So I've redesigned the vine system, and it looks very similar to this way we grew in Kent. Whilst on Ascension, I applied to become a plant protection officer, diagnosing pest and disease in a lab, working with this amazing character, Noel, who was self-taught and could mount thrips. We went back to moths, and the Americans paid for us to do a two-trap saluter survey, and they said, if you do this survey, you're going to have to not only survey the moths, but you're going to have to look at their genitals. It's a taxonomic thing. So they gave us the training via the University of Florida, and they said, this is, oh, it's gone, hang on. There we are. This is the genitals we're looking for. These are the genitals we're probably going to find, the pinworm. And they said, take loads of photos and send them to us, so we did. I had a 2-year contract, and then after that I became a garden centre manager whilst I applied for this role here. And what's really pivotal in this slide here is a nutrient deficiency trial, and we're using chemicals in that nutrient deficiency trial that were in those cosmetics that I put on the shelf in the supermarket, that I developed into face creams in my cosmetic chemist role, that I then used in hydroponic fertilizer solutions on a remote island in the Atlantic, that I then used to clear insect genitals in a lab on the Cayman Islands, and now I'm using them here to show MSc students what nutrient deficiencies look like in different species and genuses of crop. And that's it. Thank you.
Thank you, Matt. Another huge variety of roles and travelling around the world. I'm certainly feeling inspired to go do science abroad now after the last 3 talks. The last of our flash talks today is Rebecca Plater. She is a desktop support engineer here at the NBI. Please welcome Rebecca.
Buttons. Good morning. Are you a robin or a blue tit? Is my question today. Some of you might remember back to the last century when people used to get milk delivered in bottles to the doorstep, and it would have this lovely layer of cream that would settle out on the top. A lot of birds discovered that this provided a really good source of energy for them. And when the dairy companies put foil caps on these milk bottles, some birds managed to work out that they could still reach the cream by pecking a hole through the lid. I'm going to come back to that in a moment and just talk a little bit about me and what I do. So I'm part of the computing department, and we support all the 4 institutes on site. There are 6 of us desktop support engineers, and there are about 1,500 members of people that work here. There's about 2,500 computers, laptops, printers, and countless mobile phones. And we support everyone with all of those things. And that's the computers in your offices and in your labs, so on the desks, hence desktop support. Here's some pictures of things I might do in a typical day. Upgrade a computer, switch to a modern boot mode, install a computer that goes with an instrument, clone a computer, because you like cloning, set up a brand new MacBook, and connect a port to the switch so that you can have a live network port in your office. So a lot of these things, we've got processes we follow. It's comparatively straightforward when you know what to do, and you just follow a sequence of steps. Meanwhile, people will come into the office with a question like, can you help me change my password? Can you connect my phone to the wireless, please? So we're helping with lots and lots of different things. Sometimes we encounter a problem we've not seen before. And that's where it really helps that we work as a team. And there's 3 ways that we do this. So the first way might be, there was an issue with trying to log into emails, and it just kept going back round to the login screen for the person I was helping. But I asked my team, and Jordan said, well, I saw this last week, and what worked for them was I did this. I did that for the person I was helping, and it worked for them as well. So remember that one for next time. Sometimes we see something that none of us in our team have actually fixed. And we had an example of this with Acrobat Pro recently, and it wasn't combining the PDF files together for people. We had several calls about this, all describing the same set of symptoms. And so we all we tried different things between us. We all tried different things until we found the answer for that one. And another example, I was installing Windows 11 on a computer, but it kept getting stuck. I kept getting stuck in the same place. And I went through all my usual troubleshooting steps. I checked all the basics. I asked my team. None of us had seen it before. We didn't know what to do. So we got help from one of the other teams. And for this particular type of problem, I knew it would be our infrastructure team that could help with that. So I spoke to David. He had a look. He fixed it. But also, crucially, he realised it was something I could do. If I knew how to do it, I could do it. So he gave me the information, told me what I could do if it should happen again. So in each of these cases, because we worked together, instead of each of us having to figure it out separately, we got to the answer comparatively quickly. And I know what you're thinking at this point. What's this got to do with birds and cream? Well, I think it's a really good example of showing us how much benefit you get when you work together. No. And that's because robins are solitary birds. The odd robin here and there worked out how to reach the cream of this milk. But by contrast, the blue tits spend some of their lives living in a flock. And whilst they're in this flock, they've got an opportunity to learn from each other, and they can share some of the behaviours and skills that they've picked up. And this was so successful that by the 1950s, the entire population of English blue tits knew how to steal the cream. So, my question to you today, are you a robin or a blue tit?
Thank you, Rebecca. That was great. I think we can all agree. Yeah, be more blue tip. Be more like our desktop support engineers here, who I'm sure we're all very, very grateful for. I certainly am, coming to them with sometimes very trivial things that I'm not smart enough to figure out. So thank you. Appear to have done my job a bit too well. I kept timekeeping a bit too strict. So we're actually going to open the floor to questions for our Flash Talk speakers, and I believe we've let people online know as well, so if anyone online has any questions from the last 3 Flash Talks, please go ahead. Let's start in the room though. Down here.
Thanks. My question is to all the speakers. What's your top tip for the young people in the room for their career?
My top tip is that you're smarter than you think you are. And so, like, imposter syndrome is something that I think a lot of people in this room, regardless of their career stage, will say that they have. You really just kind of have to push through it and laugh in the face of that and wait for someone to catch you being an idiot. And what you'll realize is they don't, because you're not.
I think it would be ISO accreditation. And that commitment to sort of continuous learning, continuous improvement. And if you work somewhere where there isn't that ethos, then perhaps think about trying somewhere else.
Yeah, I would just say, if you find something that interests you, try it.
Thank you all. Any more from the room? No, I can certainly relate to that imposter syndrome thing, standing here right now with all these eyes on me. Yeah, you just got to give yourself a talking to sometimes, go do it. And it's actually not as bad as it seems at the other end. Have we got any questions online for our Flash Talk speakers? Okay then, thank you again. So, I'm going to... So that concludes this morning's session. We're coming back again at 2 p.m. So I hope you enjoyed this morning. Just join me one more time in thanking all of our speakers, and we reconvene again at 2.
So the networking session is over, but the science continues. Welcome back, everyone. My name is Andy, and I'll be chairing this afternoon's session. Our speakers are going to be continuing to share their exciting and cutting-edge science with us. I'm just so pleased that there's so many of us, both sitting here in the auditorium and also tuning in on Zoom. So we're going to be in reverse gears this afternoon. We're going to have the flash talks first, and then that'll be followed by 2 longer talks. With the longer talks, we'll incorporate a bit of time for questions. So if you do think of a question, please be brave, put your hand up. There's really no such thing as a stupid question. And the scientists and speakers are going to love it that you're engaging with their science. Okay, so it's time to buckle up, folks, as we take a glimpse into the journeys of 3 scientists that have achieved senior positions, but with very different roles here at the Norwich Research Park. So first up, please welcome Oliver March, who's head of molecular biology at Tropic.
Okay, good afternoon. Yes, so my name is Oliver March. I'm the head of molecular biology at Tropic, which is a medium-sized company here on the Norwich Research Park. Thanks for inviting me today. And I would just like to tell those of you how I got here, basically. So, to get here, where I am today, I followed what I like to call the Velcro approach to choose my career path. And what I mean by this is finding the areas, particularly in science for me, or topics that I find it very easy to think about, I enjoy learning about, and I find that I can happily sink hours into without spending any sort of effort at all. And for me, these are the kind of skills, techniques, and fields that I want to build my career around. And I factor this in at each stage of my development. So, for me, it actually all started back when I was a kid. So I've been fascinated by genetic engineering ever since I saw it being used on film, in one of the first films I ever watched. And this fascination with genetic engineering stayed with me as I chose my science A-levels and as I undertook an undergraduate degree at Newcastle in biochemistry. So at the same time that I was doing my undergraduate, a revolution was shaking biological sciences, which many of you will have heard of. So genome editing. This incredible technology was enabling scientists to modify genomes without introducing any foreign DNA. And with a precision like never before, tools like CRISPR had the power to correct genetic diseases, improve crops, and revolutionize synthetic biology. Recognizing instantly, as soon as I read about this, that I wanted to know everything I possibly could about this technology, I undertook a PhD and a postdoc focused around using genome editing to correct life-destroying, blistering skin diseases. And this was a journey that took me across Europe to work in Austria in a well-renowned disease research center. And I was working particularly on trying to generate gene therapies using these genome editing tools for patients. However, while I was here, again, following the Velcro approach, I realized that my true passion lay with understanding exactly what was happening at a molecular level with genome editing. and that this could form the basis of my dream career in science. And at just that time, an ideal opportunity became available here in Norwich on the Research Park, using genome editing to solve some of the biggest challenges facing the world at Tropic. So on the face of it, this could have represented quite a big career change for me, moving from therapies to crops, so moving from humans all the way into bananas, coffee and rice, which is what we work on. So what drove me to work in tropical agritech? So for me, it was the sales pitch. So the population of the world is going to dramatically increase over the next couple of years, and the food demand is going to increase at the same level. And this is going to be particularly focused in areas in the tropics. So although the UK can expect to maybe by 2050 see an increase of about 8% of the population, the world itself is expected to see a rise of about 22%, and in some areas like Kenya, Guatemala, Philippines, some of the areas that we target our key products to, the increases in growth are going to be huge. So Tropic was set up, the mission was to try and enact some change in these areas. So our mission is to create sustainable agricultural solutions which maximize the productivity and the prosperity for the growers in those areas who need it most. So, who is Tropic? We're a pioneering agricultural gene editing company with a distinct focus on the massive untapped tropical agriculture market. We actually were founded in 2016, back when we were sort of 4 or 5 people, and we've now grown over the preceding 7 years to be over 160 business and technology professionals. So that's scientists, that's our support staff, that's our business professionals, and we focus on 3 major core crops. So coffee, which is the most consumed beverage after water, bananas, which are the most popular fruits, and rice, which account for over 20% of the consumed calories in the world. And ultimately, what we're using is genome editing technologies to try and solve some of these big challenges. And what I like to boast about, in my area at least, is that our best-in-class molecular biology platform empowers us to make minimal changes in the genome, no foreign DNA at all, with maximum impact in terms of the traits. And we do this by maintaining our highest integrity as a seed company. And excitingly, in the next couple of months, years, we're working to commercialize our first products. So moving all the way from that R&D phase into something that's in the market, which hopefully you should see soon. So what do I do at Tropic? So initially, I joined the company back in 2019 as an entry-level scientist focused on molecular biology using these genome editing tools. Gradually over time, I took on more responsibility, and then I found a new passion of mine. So not only the genome editing, the genetic engineering, and the molecular biology, but actually people management. So I've now taken on the role as the head of molecular biology at Tropic for the last 2 years, and I lead a diverse group of specialist scientists who support Tropic's efforts to develop all of our novel varieties at several key stages of product development. And if I go through it quickly, these are from using whole genome sequencing and tissue-specific gene expression to enable us to really identify which core genes we want to hit to develop our target traits, using the latest genome editing technologies to target that single base pair in the genome out of about 1,500 million base pairs, using the latest high-throughput molecular analysis to identify our target crops, and packaging all of this up in submissions to regulatory bodies all over the world. So, I now encourage all of my team to follow their passion in science, like I did, to find their dream role. And I would just like to finish by encouraging all of you here who are considering a career in science, or those of you who are in science, to really follow this model as well. And at Tropic, we're always looking for passionate individuals to join us, grow with us, and collaborate with us to tackle these challenges together. Thank you.
Okay, thanks, Oliver. Fantastic insight into using some brilliant technology to do some great science. Cheers. Okay, next up, we've got Dave Baker, who's head of sequencing at the Quadrium Institute. Over to you, Dave.
Good afternoon, everybody. So, yeah, I'm currently head of sequencing at the Quadrium Institute, but this is my journey. So, as James sort of did earlier, talked about his early academic career, basically I didn't do very well at school. I actually came out of school with just 1 GCSE, and I didn't really know what to do, so I went to the local college. I did a diploma in engineering, first diploma, and then a second diploma. And after getting my diploma, I went off to university to do mechanical engineering. And at that point, I didn't know where to go. I thought, why don't I try and become a teacher? But I couldn't because I didn't have GCSE English. So I went to night school and essentially got some A-levels at the same time. So I went off to do teacher training all the way from North Devon, where I was brought up. I went via Oxford Brookes University. And then when I started teacher training, I absolutely hated it, didn't enjoy teaching at all. And it was like out of my comfort zone. So I basically looked on a map, I saw Norwich Research Park, and so I wrote a handwritten letter to the research park and got a phone call from Jonathan Jones, who at the time was the head of the Sainsbury laboratory. And then I transitioned through to all of them at one time or another. And now I'm finally at the Quadram Institute. So this is sort of pictures of me throughout my time. So I was at the Sainsbury lab for about 7 years, doing old school Sanger sequencing. And then in 2002, sort of morphed over to the John Innes Centre and developed mutation detection technologies. And then in 2009, TGAC was a brand new institute, and I was 2P'd or transferred into that institute, into their DNA sequencing, which was next generation sequencing. And then in 2018, I moved over to the Quadram. So I've done all 4 institutes. So essentially, this is just an image of all the DNA sequencers that I've made libraries for and run and maintained, so it's quite a lot. So obviously, it's quite a lot of experience in DNA sequencing. So over my time, which is almost 30 years, this December, I've traveled all over the world. So it's, in terms of working in science, it's a great opportunity to travel around the world and see different places. And actually, this Sunday, I'm flying to Zimbabwe, which is an interesting. So this is 2018 and 2020. I went out to the Gambia to help in 2 training workshops. And then in 2022, I went out on my own to Ghana to implement nanopore sequencing within the University of the Cape Coast in Ghana. And the other big impact story that I was involved in is basically coronavirus. So back in 2020, when COVID started, the method that was used for sequencing the variants was limited to 24 samples. And I developed methods within the Quadram to do sequencing of the COVID virus to a high throughput method. So in the end, during the pandemic, in my particular lab, we sequenced over 87,000 COVID samples, looking at the variants across the whole pandemic. And then it eventually got me my first authorship in a publication of the method called Corona Hit. And this is just the team that I've had over the last 6 years, so it's just not myself. So thanks very much. Hopefully it wasn't too quick.
Okay, thanks, Dave. Much appreciated. Fantastic career here at the Norwich Research Park. And finally, I'd like to welcome Neil Shearer, who is the Laboratory Information Management System Manager at the Earlham Institute. Over to you, Neil.
So here's some questions for you. How many of you know what you're going to be doing or you want to be doing in 20 or 30 years' time? If you're currently working, how close is your current role to the one you envisaged you would be doing when you first set out on your career? Well, when I left school at 16, I had no plan. I had ideas of joining the RAF as an aircraft technician, but I failed the medical. And after that, I really had no direction. And so I worked in a variety of unconnected jobs over the next 7 years. I worked as a pig farmer, and I tried my hands at vehicle mechanics and being a post office clerk. I spent 2 years as a trainee butcher, and I worked in this factory making these handmade roofing tiles. I even tried my hand at grave digging. Although these jobs are largely unconnected and might seem irrelevant to what I do now, each role taught me something about myself and helped me focus in on what I wanted to do next. At the age of 24, I was out of work and frustrated with my life, so I returned to full-time education. And this is me
sitting by the fish pond that I built at college. So I completed a life sciences access course and discovered fascination with genetics. So driven to understand how genes are switched on and off in different times and different cells and in different tissues, I enrolled at the University of East Anglia to study microbiology. This was followed by a PhD in bacterial genetics, and then 15 years after that, 15 years of being a research scientist across the Norwich Research Park at the UEA, John Innes, and there's a John Innes, and the Institute of Food Research, which is now the Quadrum. During that time, I learned many incredible mechanisms by which gene expression is regulated in bacteria and plants. At the age of 24, sorry, I've already said that bit. However, whilst I enjoyed learning about science, I found that focusing in on very small areas of research a little frustrating, and increasingly I found more motivation and reward from helping others achieve their research goals. So in 2014, I re-evaluated my career goals. I gained a project management qualification, and I joined the genome sequencing team at the Earlham Institute. Initially, I was involved in managing sequencing projects, and later my job evolved, and I now manage the group's Laboratory Information Management System, or LIMS, and I'll come back to that shortly. So we process tens of thousands of samples through our facility each year, and without dedicated software to manage those samples and the related data that's produced during their processing, it would be impossible to keep track of all the samples, and that would inevitably lead to errors. And that brings us to the LIMS. A LIMS is computer software that helps us and is designed to and helps us manage all the samples and data. It helps us track and record everything that happens to the samples, and allows us to direct the samples down the relevant sequencing pipelines. This allows us to accurately and effectively manage our workload and provides traceability and supports reporting and troubleshooting. So what do I do as a LIMS manager? Well, I assist users with day-to-day inquiries, and that's the favourite part of my job, along with working with them to develop new workflows. I'm involved in training staff to how to use the software and providing written user guides to further support the team. I spend a lot of time testing and implementing software updates and new features. And I also undertake regular maintenance tasks to ensure that the data records are kept up to date. There's always something new to do, and currently I'm working with other members of the group to establish a automated instrument pipeline that will streamline our sample QC. So I didn't train specifically to be a LIMS manager, but the transferable skills and attributes that I picked up during my career underpin everything I do. That, along with my laboratory work, is essential to everything that I do. I've highlighted coding here because, perhaps surprisingly for a software job, I don't actually need to code. However, as we look to integrate the LIMS with other software that we use, then coding would be a useful asset, so it's something I'm looking into. As I come to the end of my talk, of course, no one gets anywhere in life without a little bit of help from people, and throughout my career, there have been people that have motivated me and that have helped me, and some of them are in the audience today. So finally, I hope you've learned today or heard today from my talk that you don't always need to have a career plan. As long as you consistently learn about yourself and are willing to readjust when you take the wrong path, then you will succeed. I also hope you've learned a little bit about what LIMS software does and what a LIMS manager involves and requires. If you want to know more about LIMS or being a LIMS manager, or perhaps how to get rid of a body, then please come and speak to me after the talk or send me an email. Thank you for your attention.
All right, fantastic job, Neil. Thanks for giving such an honest account of your journey to your current career. So I think it's clear from our flash talks that while there is a well-trodden route into science, it's not the only path to get there. And it's never too late to follow and pursue a career in science. I hope that we've also convinced you that because research science requires such a broad range of expertise, you don't need to be a scientist to contribute to science. So our next speaker studies a type of virus that's so abundant, scientists find it in pretty much everywhere they look for it. It even lurks inside the human gut. But here's the thing, it's not after human cells, it's seeking out bacteria. So what do these viruses look like? What happens to the bacteria that are infected? And can we actually use these viruses to improve human health? So to give us a lowdown on this and answer all these questions, please welcome Evelien, a group leader at the Quadram Institute, who will give her a talk entitled Bacteriophages, the Friendly Viruses.
Hello, everyone. My name is Evelien, and I'm a group leader next door in the Quadram Institute. As the previous speakers did, I also quickly want to talk to you about my background so that you know where I'm coming from. So I'm originally from Belgium, and I studied at the University of Leuven, and I stayed there for 10 years doing all my degrees. And I studied bioscience engineering originally, because when I was in secondary school, I wasn't quite sure what of science that I liked most. And the course of bioscience engineering had the most options, because you could specialize in chemistry, in biology, in environmental science, soil science, all forestry, whatever you would like. And it wasn't until I was in the last year of my master's that I really found my passion, which is bacteriophages, and that's what I did my PhD on. But when I finished with my PhD, I had spent 10 years in the same spot, so I thought it's time for a little bit of adventure. So I did my first postdoc in South Africa at the University of Pretoria. And there I was investigating bacteriophages and other viruses in desert soils. And yes, bacteriophages are also in desert soils. When I was in South Africa, I learned a lot about bioinformatics and high-throughput methods, and I took those and I applied for a job at the University of Liverpool because they were looking at some pathogenic viruses, and I thought, oh, maybe that's also interesting. So I stayed there for a couple of years, and then eventually in 2019, I started my research group here at the Quadram Institute, where I'm combining a little bit of my expertise of all of the previous positions that I had, but focused on more human health. So I've said the word bacteriophages a lot now, and I want to go a little bit deeper into what these are. But to understand bacteriophages, you first need to understand what are bacteria. So briefly, and perhaps a little bit simplified, bacteria are single-celled organisms, and they can grow by themselves. As long as we give them the correct environment and temperature and nutrients, we can grow them in the lab. And when we look at it from our perspective as a human, these bacteria can be beneficial for us, they can be neutral for us, or they can be bad. So for example, beneficial bacteria could be the bifidobacterium that you need in your gut, that babies need in their gut because they can process and digest breast milk. Or you can have lactic acid bacteria to make cheese. There's loads of bacteria out there that you never really get in touch with at all, and they really don't matter for you. For example, I have a visiting scientist in the lab right now who's looking into bacteria that live in solar Saturns, for example. But of course, there's a lot of bacteria that can be bad for us. You can get a foodborne illness with a Salmonella or Campylobacter or E. coli. You can have an infected wound. And there's loads of other bacteria that can be bad for us. We've heard about crops earlier. So one of the things that I did during my PhD is I rotted a lot of potatoes, and I looked at bacteriophages to prevent those potatoes from rotting. So what are bacteriophages then? These are viruses that infect these bacteria. In short, we use the term phages a lot. It's just because we're lazy and we don't want to say too many syllables. They are obligate parasites. What does it mean? They cannot grow by themselves. They need to infect the bacteria in order to reproduce themselves. And they look amazing. Like Andy said, they have all sorts of wonderful shapes. And this is not an actual picture, but a rendering by an artist. So what do these bacteriophages consist of? So these are the most commonly found ones. They're head-tailed, and in their head, they have their genetic material, and they have a tail with tail fibers that they use to attach to the bacteria. In simplified form, it looks a little bit like this. So you have your bacteriophage, which is much smaller than your bacteria, and it attaches, is this the laser? Yes. So it attaches to the bacteria. Once it recognizes that it has the correct bacteria, it injects its DNA into the cell. And that DNA then gets used to make new bacteriophage particles. So basically what the bacteriophage does, it makes the bacteria into a virus factory. And then at the end, when enough bacteriophage particles are made, the cell breaks open, the bacteriophages are released, and the whole cycle can start again. And of course, this is something that we immediately are interested in, because when we think of those bad bacteria, we think about ways that we want to kill them. And one of those ways that we're investigating is using bacteriophages to kill bacteria. So how do we find these bacteriophages? So it's quite a straightforward way. We grow the bacteria that we want to kill onto a plate in the laboratory, and we make a lawn, meaning that we just make sure that the whole plate is covered by the bacteria. And then we add something to it where we think the bacteriophages are in. This could be, most oftenly, we use wastewater, and we filter it, and we use that because there's a lot of bacteriophages in there. And then we look for these kind of zones of lysis. And these zones of lysis mean that the bacteria have been killed, and that's where we can start, okay, this is potentially a bacteriophage. And if you mix them into the lawn, and they spread like this, and you see the small spots, we call them plaques, and this is how we go hunt for them. Now, this is a kind of low throughput method. What we've been recently doing in the lab is kind of more high throughput analyses. So one of the things that we're doing is we do it at a larger scale. So we put, and also to investigate the bacteriophages, so we make a dilution series in a plate. This is my postdoc Hannah doing this. So we have a fancy pipette that we bought. So we take the bacteriophage solution from here, and we plate it onto what becomes a lawn, and then it looks something like this. And then we can easily compare different bacteriophages against each other. So for example, these are all the same bacteria, but there are different bacteriophages. And you see that this one doesn't grow very well. You can see that this is always a dilution from the highest to the lowest, and you can see that there's differences in the quantity of bacteriophages that are in. There's differences in how they look, what they look like. So we can already, in a first instance, differentiate them. Why don't we use a microscope? Well, one of the things is that they're too small to see in a normal microscope, because they are only about 200 nanometer, 250 nanometers in size, so we need an electron microscope. And this is what it looks like. And this is the first bacteriophage that I found myself during my PhD, and this was 16 years ago, this was found. So again, it looks like, you know, a head, a tail, and the ones that I found didn't have tail fibers, which was different from the ones that we saw originally in the artist rendering. And so we found many of them. I have a heavy-handed click, apparently. So these are some of the bacteriophages that either myself or members of my team have found over the course of the years. So these were 3 of them that were part of my PhD. This one is actually a printed picture that's hanging in my study at home. These are much smaller ones that don't have the tail. They also exist. And these are 2 that we found here in Norwich. Now, to give you a sense of the scale, these are 30 to 250 nanometers in size. That may not mean anything to you, but we're going to go up in size. So this is what they look like in the context of a bacteria. So this is an E. coli cell, and all of these dots are this bacteriophage bursting out of this E. coli and killing it. So you can already see that this is a big difference in scale. And this picture was made by Heather Allison, in whose lab I worked while I was in Liverpool. But if you then would put that E. coli on the pin of on the tip of a pin, then that's what it looks like. So you can barely see them when you've zoomed in 85-fold times, and you need to go about 2,000-fold to make it look like the previous picture. So this is why we need to find different methods to visualize them, like electron microscopy. All right. So what I want to talk to you about now is how we and other people are looking into phage applications to kill bacteria. And this is not a concept that I've come up with. This is actually over a century old. So the bacteriophages were discovered independently in 1915 and 1917 by scientists in France and the UK. And already in 1919, they realized, hey, we can use these bacteriophages to kill bacteria, so we should do something with it. So the first described published case was on using bacteriophages to treat bacterial dysentery. And in the 30s, so this was already 1919, then it kind of was followed up with other kind of treatments. But by the 30s, it was clear that people were just using bacteriophages willy-nilly for anything. So what I told you before is it's a virus that needs to infect bacteria, but people were using it for illnesses that were not related to bacteria. So you're not going to kill a virus with another virus, or you're not going to kill a fungus with a bacteriophage. So there were a lot of kind of treatment failures because people didn't really understand how these bacteriophages worked or how certain diseases were caused. And then in the 1940s, antibiotics started to get introduced, so we didn't really need bacteriophages anymore. So they kind of fell off the face of the planet in terms of research into their therapeutic use, except for in Eastern Europe. But we had then a resurgence of phage research in the West because of the rise of antimicrobial resistance. And since the 2010s, we have in many countries across the world compassionate use cases of bacteriophages. What does that mean? That means that if all treatment options have been exhausted, a a product can be used that that it's not authorized as a medicine. So there have been a lot of cases of phage therapy across the Western world based on this. Now, one of the institutes that has been doing bacteriophages for 100 years is the Eliava Institute in Georgia. And they've just celebrated 100 years of doing bacteriophage research. And I'm not going to elaborate on this, but if you're interested in learning more about this, they have an English website, so you don't have to learn to read Georgian. And there's also a book that has been published recently called The Good Virus, which gives a really fascinating view into the history of bacteriophage research. But what I wanted to talk to you is about what we're doing now at the Quadram Institute. So we have different projects running on bacteriophages, understanding how they contribute to a healthy microbiome, understanding the phage-host interactions, because your phage has to connect to your bacteria in order to start its infection process. We have a project on treating wastewater to reduce antimicrobial resistance. We have a project on phages against foodborne pathogens, and we have a project on phages against the bacteria Pseudomonas aeruginosa in chronic lung infections. So first, I'm going to tell you about the Phageland project. So this is a project that is a big European project with 7 different partners, where we want to combine the power of bacteriophages with what's called constructed wetlands to treat wastewater, so there's fewer pathogens that carry antimicrobial resistance genes. And this is led by Hannah Pai, who's a postdoc in my group. So one of the things that she did is she made a huge collection of bacteriophages, and she's the one who developed these nice square plate assays. So one of the things we wanted to do is we want to have bacteriophages that are stable at 10 degrees, because that's the temperature that one of these wetlands works at. And we wanted to have bacteriophages that are stable for a while in sunlight, so UV exposure, to get them into what we're calling a phage cocktail. So this is when you put multiple phages together that can either treat or infect a range of different bacteria, or that prevent resistance from happening. So one of the key steps in this was creating large bacteriophage stocks. So we just have to grow up our bacteria in large quantities, multiple liters, and we're doing that in a cell maker. So we've mixed bacteria in this, we let them grow. There's oxygen, air pumping through to provide the bacteria with oxygen, and then we inject the bacteriophages and let them multiply and then harvest that. And then this is when we finally purify and concentrate that. This is what our bacteriophage stocks look like. So we have them in little vials and we keep them in the fridge. So what are we going to do with them? So we're concentrating a lot of them, and we're talking about hundreds of billions of particles into one of these vials. We're going to put that into the constructed wetland, and that's basically a set of skips where your wastewater flows through, and it's meant to be a low-cost solution that can be implemented in low and middle-income countries, where it's operational from this month. So our collaborators are in Poland, and they're currently running the baseline for the wetland, and then we're going to treat it with bacteriophages, and then we're going to monitor how well this works. So before and after phage treatment. The second project I wanted to talk to you about is called Bronchophage. And this is a collaboration with the Norwich and Norfolk University Hospital, where we are investigating the use of bacteriophages for the phage therapy of Pseudomonas aeruginosa in chronic lung infections. So we have a lot of people in the area who have chronic lung diseases, and many of these people have exacerbations, so they have extra infections that cause problems and that cause their quality of life to be quite low. So what we want to do is we collect sputum samples from these patients. So they send them by post to our institute. From those sputum samples, we isolate bacteria and we investigate whether we have bacteriophages for them. This is a very, very early stage project, but it's very exciting to get started on something where in many cases there is no viable treatment at the moment. So what we do in the lab is we get the sample in and we divide it into different aliquots to then keep. We have a laboratory management software that we do use. In this case, it's a sample management software called RedCap. And what we do is we isolate the bacteria. And we do that by streaking them on plates, and we have selective media, meaning we only isolate Pseudomonas aeruginosa that we need, and then store the isolates. So this is what it looks like when you see those on different plates. So we see the bacteria, and then we just take them and grow them up in pure culture so that we have them to then do our bacteriophage isolations with. And this is what it looked like. This is very early days in the project. We still have a lot of work to do. So what we did is, on the right-hand side, you can see here again those plates. So this is one of our lab strains that we know we had different bacteriophages for. And then we took the strains from the hospital patients, and we realized that our bacteriophages that we already had didn't work on these. So, and this is how you should interpret this. So when there's a yellow box, it means a phage, which is all the columns, is infecting the bacteria which are in the rows. So this is our preliminary test, and this is why we know that it's important to do this project, because we now know that we need to isolate new bacteriophages, because these bacteria that are in these patients are quite different from the bacteria that we work with in the lab. So the next step in our project is now building this extensive phage bank so that we can eventually, hopefully, treat these patients. So I'd like to kind of round this off by talking to you about the pros and cons of using bacteriophages to kill bacteria. So one of the pros is that they're safe, because they're viruses that only kill bacteria. They don't infect humans, they don't infect animals, they don't infect plants. They are naturally occurring in the environment, and in fact, we use the environment to isolate them. But because they can replicate themselves, we don't have the same resistance problem that currently exists with antibiotics, because each time a bacterium becomes resistant, the bacteriophage can then overcome this resistance by continuously evolving. As I showed you, they can be used in many different applications, because we've been investigating wastewater treatment, we're investigating treatment in humans, but we also investigate treatment in the food chain or in crop production. And one of the things that has become clear as more tests are being done in labs and in compassionate use cases is that we are looking at how they work well with the antibiotics. Rather than just replacing the antibiotics, we want to make some sort of combination therapy. Now, you think, oh, everything is great, let's just always use them. No, there's a couple of cons as well. And one of them is that these bacteriophages have a very narrow host range. As I showed you on the previous slide, we need to have a specific bacteriophage matched with a specific bacteria. That means that if a patient has an infection that is rapidly progressing, and so if a patient has an infection that's rapidly progressing, we might not be able to match it with the correct bacteriophage in time. So this is why currently most of the treatments that are being done in humans are people with chronic infections, so that there's time to find the correct bacteriophage. They are not always stable, and that has been an issue in the past, where by the time you treat a patient. There weren't as many in the vial that we wanted to have. They're more difficult to produce than traditional antibiotics. Traditional antibiotics are a chemical compound that you just produce and you can check as is, whereas these are quite complicated biological entities. Producing them, and this is something that we realized in the lab when we were doing our liters of bacteriophages, it was quite a long and time-consuming process. There is still resistance development, so we still need to make sure that we do our laboratory experiments correctly so that we know that we can overcome this resistance development. And currently, they are not approved for use on a regular and routine basis in the UK or in many other countries. So this is something that people across the world, scientists and regulators combined, are kind of working on to get this therapy or applications in many different fields approved. So with that, I would like to thank everybody. This is not just my work, this is a work of a lot of people. So I'd like to thank everybody who's in my team, everybody who's worked with me on these projects, particularly external collaborators and people in the Institute and the Human Studies team, and my funders as well. Thank you.
Fantastic talk, Evelien. Thanks very much. It's really good that we've got these phages that we can potentially use alongside with the traditional antibiotics. It's really good news. Do we have any questions in the audience? Yeah, right at the front here.
Hi, thank you. That was a lovely talk. It's really interesting. I was just wondering if you had any thoughts on delivery methods for phages for patients.
Yeah, so it will depend on what we're treating, right? So if you're treating a wound infection, that will be a topical cream or a gel. If we're thinking about the chronic lung infections, this will probably be a nebulizer, inhaler. There's instances where people have been given I don't know how to prescribe it because I'm not a clinician, but when they were infected wounds, then they were rinsed with the solution, and there's been intravenous use as well. So it's basically whatever you need to do to make sure that the bacteriophages get to where the bacteria are.
Seems quite versatile, thank you.
The back there.
Thank you so much for your talk. Is there any chance that you could possibly go over the lytic cycle again, just quickly, if that's okay?
Yeah. Oh, no, but yeah. Okay. So we start with bacteriophage floating around in whatever environment we are, and the bacteria as well. They need to find each other, so the bacteriophage uses its tail to connect with the bacteria. It is what we call adsorption, and then it injects its genetic material inside the bacterial cell. And then that DNA gets transcribed into proteins, and that eventually gets made into more bacteriophage particles that are assembled in separate pieces. At the same time, the bacteriophage DNA is also replicated, and then it all kind of assembles into the cell into full particles. And then, let me go one further. Yeah, so then they express an enzyme that lyses the cell, so it bursts open, and then you have new fully formed bacteriophage particles spreading, and then it all starts again.
I think one more question from the audience, and then we'll head to the online questions.
Thank you. Is evolution of the virus and the bacterium as they fight one another, or possible recombination with bacteriophage in the environment. Are these obstacles to licensing?
So yes, there's actually quite complicated dynamics that go on in nature between bacteriophages and bacteria. And if bacteriophages were 100% successful in killing bacteria, there would be no more bacteria. And if bacteria were 100% successful in developing resistance, there would be no more bacteriophages. So in nature, you have a balance that gets, that keeps going. But when we try to treat people or treat in environments, then what we want to do is we want to disrupt that balance. So we add many, many more phages than in a natural case would be. So 100, 1,000, 10,000-fold more. And then you can see that the bacteria either get eradicated or, for example, in the case of the human body, the immune system can then take over. So there are instances where it could be a problem if there are some bacteria that have incorporated a lot of defense systems. But in many cases, we can still find a bacteriophage, or if the bacteria incorporates that many defenses and mutates itself that much, it becomes less fit to be causing an infection. So then again, the immune system can take over.
Thank you. Sam, what do we have coming in remotely?
Lots of interest online. In fact, you've already answered the first one, so I'm not going to ask that. So Edward Ingalls asked, is there anything to suggest the phages can be used on fungal infections? And if so, is there anything to say that they'll be effective?
So on fungal infections. So no, they will definitely not work on fungal infections because a fungus is a completely different organism. Now, fungi have their own set of viruses. And I know that some people are looking into using those viruses, but it's a bit more complicated because these viruses are a lot less easy to culture. So, yeah.
Okay, thank you. It's nice to have a definitive no to an answer to a question rather than that. And we've got a question from John Thankersham. Do you think this could be a viable frontline treatment for bacterial infections across the body in the future, or would it be too dangerous or expensive?
For example, in Georgia, they are already doing that. So in Georgia, they have over-the-counter bacteriophages available as a first line. But the problem is that antibiotics are much more broad spectrum and bacteriophages are very narrow. So you're likely not gonna cause any harm, but you might not just do any good if you don't match the bacteria with the bacteriophages that you treat with. So it could be, depending on how bleak the future is in terms of the availability of antibiotics, it could become a frontline treatment. We don't really know.
Thank you. And one more from Helen Dorrell. Thank you for an interesting talk. With reference to the human microbiome, have you come across phages being harmful by carrying antibiotic resistance genes into bacteria where they can be replicated by the bacteria?
So there's definitely harmful bacteriophages out there from a human perspective, that is, because one of the things that I didn't show is that there's another cycle that some bacteriophages can use is a lysogenic cycle, and that means that they incorporate their DNA into the DNA of the bacteria and then get inherited. So we want to stay away from those bacteriophages and only focus on those that we call virulent, that only have the lytic cycle. But the ones that are we call temperate, that have the lysogenic cycle, that integrate themselves, they are some of the most responsible for some of the worst diseases. For example, the cholera toxin comes from a bacteriophage. The Shearer toxin that causes E. coli to become very enterohemorrhagic also comes from a bacteriophage. So when I say that these are friendly viruses, it's actually only a subset of them that I'm using for to make it simpler. But yeah. Although antimicrobial resistance, we don't see it that much.
Brilliant questions, folks. Let's thank Evelien once more for a brilliant talk. Okay, so when I was reading the information about our next speaker, can you hear me okay? Yeah. I was Googling the scientific names of the creatures that she was interested in. And pretty soon my browser's filling up with close-up images of mosquitoes, fungus gnats, and assassin flies, all staring down at me from my computer screen. Anyway, by the time I got halfway through the information, I was swatting away at non-existent flies. I think it's fair to say that flies in general are not particularly well liked by humans. But those that have looked beyond the negative aspects have discovered the truly fascinating lives of these insects and the variety of crucial roles that they play on the planet. One scientist that has followed her passion for all things fly is Dr. Erica McAlister, the principal curator of flies and fleas at the Natural History Museum in London. As well as managing the museum's collections, Erica teaches and studies entomology and has written several highly regarded books. She regularly contributes to science communication, including presenting radio shows for the BBC. Today, we're extremely lucky to have Erica with us. And with no further ado, let's welcome Erica up to the stage to present her talk, Inspirational Insects, Why Flies May Be the Answer to Everything.
Thanks. Why start a talk telling me you're swatting flies? Generally, I know how to get rid of your body. I'm just saying. I'm not a geneticist, I'm not a molecular biologist, I'm just a person, just a female, standing up here telling you about, oh, this is quite fun, why I like these things. Now, they are some of nature's best creatures, there's no doubt, just look at them. Every single one of them is really cute and really fluffy, and you can see why flies have got an international reputation of being loved by all. So we have some amazing things here. First off, never trust a dipterist. Being studying flies, what you're seeing is a cute little face there. Some of you in the audience will know me and know never to trust when I put a cute little face up, because what actually you're looking at is the anal spiracles of a maggot. So you're looking at its bum, but that's fine, that's fine. And then you've got all these beautiful bat flies, and what I love about flies is they just fly in the face, quite literally many times, of what you expect from them. And I'm really lucky. I get to travel all around the world. I already told some people today, all my exotic sites. Yep, there's me hoovering a pig in Hounslow. And that's a career highlight. Unless you've hoovered a pig, you have not lived. This is me with a llama. It was a particularly ugly, goofy llama, but it quite liked me hoovering flies off it. And yes, because I study flies, I do hang around feces. This is me hoovering Titum arum, and I don't know any people who've got, I've hoovered off dead things a lot, and I came close to vomiting when I was doing that, and it was in Kew Gardens, and there was hundreds of school kids, and they were like, oh, what's that, what we're doing? And I'm like, please don't vomit, this is the worst career. It's an amazing thing, but I do get to travel exotic places, and one of the things I do is I describe flies. Now, never upset taxonomists. We didn't start it, Linnaeus started it by naming lots of species of weeds, what he called weeds, after people he hated. Now, I've described lots of flies after people I love. This fly here is from the Caribbean. Thank you, taxpayers. I spent many months out in Dominica collecting flies, drinking rum. It's fantastic. But the woman who organised it, she spent weeks out there trying to sort it all out for us and whatever. So as a thank you, I described this fly after her. And why I specifically use this one, because 7 weeks in the field, she had really hairy legs. And I gave it as a wedding present. She loved me. But I, like many of the scientists, there's 350 of us research scientists at the Natural History Museum. We go all around the world, we're doing all sorts of fantastic things, and we bring them back to the building of what the public refer to as bones and stones. Okay, they just go through the museum and go, I want to see dinosaurs. And we're not allowed to physically hit children. So we go behind the scenes and we're like, it's okay. What you see on display is 1% of the collection. There is 80 million specimens behind the scenes. It spans the breadth of the entire world and beyond, but it also goes back millions and millions of years, although most of my flies are a little bit younger than that. And what I do is I take them back, and our collection of flies, it's the biggest in the world. It's great. It's all mine. Apparently, it is yours as well, but it is really mine. And we have about 2 to 4 million flies. It's a big, big guesstimate because we have thousands of jars. Yeah, if you want to come and count them, go ahead. So we have them there, we bring them back. But why should you care about flies? Like, it's the biggest question I get asked by everyone. It's like, what's the point? People talk about swatting them pretty much straight away. And I'm like, oh, it's a bit harsh. I don't go in your house and kick your puppy. So please do not go out there and hurt the flies, because actually the flies are very, very useful. So first of all, I thought, because maybe some of you don't know what a fly is. So there's many names that we use for flies: dragonflies, house flies, scorpion flies, all these sorts of things. Some of those are flies, some are not. To be a true fly, a Diptera, Diptera, 2 wings, okay? Now, not all flies have wings. They're not called walks, before we go down that line. But the adult flies, generally their basic body plan was to start off with 2 wings, 1 pair of wings. You then have suctorial mouthparts. So another thing that people say is, oh, I kept getting bitten by these flies. And I'm like, no, actually, that's not correct. You would have been sliced open, lapped upon, pierced, stabbed, all of those things, but they can't actually bite you. This seems to be lost in the translation a little bit. But also, they have these halters, and these are these balancing organs. These are the proprioceptors, and enable them to do all sorts of crazy stuff, doing amazing aeronautical feats of amazingness. I say amazing a lot with flies, I'm sorry. So when people talk about birds and their fantastic feats of migrating thousands of miles, flies are doing that. They are most amazing aeronautical engineers. But that is just one part of the fly. And this is where we think, you know, mammals, we're really quite dull. We're born and we just get bigger. There's nothing more fun in our life cycle. But flies, they go through what is called a complete metamorphosis. So often they turn up as eggs. I say often. Some go through very similar life cycles as us in the fact that they give birth to live young. But to anyone who's given birth in the audience, some of these flies give birth to something that has a mass bigger than them. Yeah, ooh. Oddly, they only give birth to 1 or 2 maggots, because why would you? Oh, anyway, I'm going to move on before I cry. We got the egg stage, so this is the rapid division. We're growing and growing, and then we have the larval stage. And this is really important. What do baby mammals do? Cry, eat, and that's it. Pointless. What do baby bees do? They just eat. They may cry, I don't know. But again, they're not doing anything. Baby flies are out there working. Okay? They're out there already helping the greater society. They're already helping the planet to look after each other. This is why we need to give more voices to the little unspoken maggots. And then we go to the pupal stage. And this is, I think, arguably one of the finest things has ever happened. Because within here, within this hidden stage, you get this almost complete reorganization of tissue. The idea of being able to change from one form, this consuming, eating, lazy stage, to this one that can locomote and does exactly what that picture is showing you, the most important thing, producing the next generation. So the fact that you've got all of this, you've got all these different parts, enables flies to actually dominate in so many different types of the ecosystem. And here we go, this is a speciescape. So this is a representation of how many species so far have been described to date. Look, there's the little pointless, pathetic mammal. Ta-da! There's little fungi, although I'm not sure what species are with fungi. I've already had this discussion today. Okay, and we've got some nice arachnids doing well, plants doing well. Ta-da! Look how many insects have been described. And I like this because we got the fly as it. The fly is the best. So to date, we think there's about 6,500 species of mammal, whatever. There's more species of fly in the UK than there are mammals on the planet. There is over 165,000 flies described today. A lot of them. And they're everywhere. And in being everywhere, they're able to do all of these things. Okay, they are the most ecologically diverse. Their functionality is amazing. They can eat things inside out. They can eat things outside in. They pollinate. They are decomposers. They do all of these things. And this is what we don't give them credit for. So here's some of the beautiful hirsute heroes that we come to every day. We're face to face with these. This is my little garden in South London. Okay, it's nothing special apart from look at them. They're amazing. I absolutely, these lovely fluffy things. When they go on about mammals being really cute, hello. Look at these. And these are really important. And what we're learning more and more about these every day, you all recognize a bee fly? Pardon? Thank you. So this is one of the best animals in the UK. This bee fly, what we're still learning about it is its life history and how important it is. So for example, look what it can do with its proboscis. It can massively expand it. So this is really cool because these turn out to be quite good pollinators, and they're pollinating long tube flowers. They're not as good as some pollinators. Check out this bad boy. If that was a human, your tongue would be 6 meters. Okay? Just saying. And this is amazing. This is Megasterynchus longirostris. This is found in South Africa. This is found in the Cape region. What is the Cape region really good for? Plants. Why? Because they've co-evolved with all these fantastic flies. There is a lot of Ericas growing in the Cape region, which I'm quite pleased about, and I'm pollinated by flies, which makes me feel very happy. So we've got these amazing adaptations. Remember, there's no muscles in this, and this is quite useful to think about in a minute. We've got these tiny little flies, okay? You know these all as biting midges. Most of you hate them. You think they should all be destroyed. I'm like, oh, but if you destroyed them, you destroy that. Oh, now I hate chocolate, so I'm totally happy for chocolate to disappear, but most people don't. So of the 25 or so described pollinators of chocolate, there's one tiny ant, one tiny moth. Most of them are these tiny little no-see-ums, these biting midges. Really important. Oh no, those mosquitoes that everybody hates as well. Okay, they're really useful in pollination as well. We've only started to look at them. In the Arctic region, there's 4,000 described species of insect, 2,000 of them are flies. There's some guild pollinators. You get rid of mosquitoes, you lose some plants. And their larvae, birds love them. When I was doing my PhD, I was working on wetland entomology, and the head of the department was like, oh, you're just doing bird food. And I was a little annoyed, but she had a point. And it's really important we have all these larvae to help all these different food cycles. And going back to those mouth parts, those things, if you ever get some time, just do watch a mosquito feeding on you. It's quite fascinating. Nope. I've got my people, it's all right. And what these do, she's able to sniff you out, okay? So she takes a series of cues to find you and sniff you out, and then she has sensory capillary on the end of her proboscis, which she stabs through your skin, but not directly. She basically has a pneumatic drill. So she's able to penetrate your skin at 400 times less force than if she was just a ram in her proboscis. Amazing. That's got smart needles written all over it. Underneath, when she's underneath your surface, she can move her proboscis round and she sniffs out your capillaries. How cool is that? Now, this is not just cool for me, he's like weird, but this is really important for medicine. So think about doing invasive surgery now and able to ignore or bypass some of your very sensitive tissue. It's really cool. So I got quite obsessed by all of this. They laid me on radio. I got highly sensitized because most of my conversations were not appropriate for Radio 4. And then we wrote a little book about it, me and my wonderful producer, Adrian Washburn, who told me off more times than any parent either parent I had. So I'm just going to talk about 3 little stories, highlights from the book, about flies, which I really like. And the first one is me playing with feces again. I mean, what sensible child does not end up playing with feces? Love it. But just think about feces for a second. When they say, oh God, there's so many flies on feces, just say thank you. Because imagine if there wasn't flies on feces. Imagine it was just covered in bacteria, some of it good, some of it bad. Oh, it's slowly decomposing. But all of that feces, just think about how much you defecate. Maybe not too much, but you have a lot of this. If we did not have flies decomposing, you'd be swimming in a quagmire of feces. Not just the feces, you've got decomposing bodies, yeah, that haven't been destroyed. They haven't been recycled back into the environment. So not only do you have this feces, this river of feces, I don't think I've ever used that expression before, this river of feces with all these dead bodies of long-lost relatives floating past you. So we have to thank the flies. And actually, especially now, we have to thank them because of climate change. So we have eaten insects. I love I'm going from feces to eating insects straight away. Maybe I should have thought about this. But we eat a lot of insects globally. So at least 2,000 species probably is eaten across the world. We are a little sensitive in the UK, which is funny because all of you are eating insects whether you like it or not. Food Standards Authority let loads of different types of insect get in your food. Yay! But most of those, you'll be glad to know, are little bits of beetle. Sadly, only 2% of... I love the fact I'm saving flies, and yeah, I'm encouraging you to eat them. Apparently, eating maggots is something that we don't like doing. So let's go back to our feces-loving things. Oh no, hold on. I'll move on to burgers first. Just as I'm talking about feces, good luck eating dinner tonight. We have a climate problem at the moment, which we know is really bad. So we started to think about going back to insects and actually producing lots of viable alternative protein, this insect protein. Because eating beef is a really bad thing. It's a humongously bad thing. So it's the CO2 equivalent, 2.5 million beef burgers a year. Apparently that's like 7.8 burgers a year, which I think is still quite a lot. You should see the American statistics. That was vastly entertaining. But that is the equivalent of you driving one car 43.5 billion kilometers. Apparently, that's 1,000 times around the planet. So just think of the CO2 impact of that. And it's not just that. We've got to think how we feed these creatures, okay? So we're not just thinking about the environmental cost of the CO2, but also all of these. And the land, we know how much is being deforested. We also know we have a water supply problem. Okay, look how much water a cow needs. Look at the little insects. They need nothing. And you can eat most of the insect, okay? There's hardly any waste associated with it. This is great. And there was a dude, Charles Raleigh, he's the one seriously jealous of that moustache. I'm growing one, but it's not as fast. He was a British entomologist, and he was the one who first started getting involved with this. He headed across to America. Anyone in the room who's ever had their parents tell you that they're really disappointed about you, his mother, on her death certificate, died of disappointment. That was a genuine thing. I don't know how that happens, but whatever. Now, he was being asked by lots of people to, the farmers were complaining, beehives had loads of, they were saying all these things were in there eating these baby bees and it wasn't good. And he realized they weren't baby bees, but they were Matia lucens. This is the black soldier fly. And he was like, oh, okay, hold on a minute. What are they doing? And they were eating the waste. They were converting the frass that all the bees left behind into usable protein, i.e. themselves. So we went and looked at a protein factory. So this is underneath the railways of London. And these dudes had completely automated a system of producing black soldier flies on the waste material, the vegetables from all the restaurants in the area, and they were able to grow. Look, here's my producer recording flies, you know, making more flies. And these are the baby flies, and what they were able to do was to automate it so that they could produce flies with the correct amount of protein and fat for the different diets of the animals, such as cats or dogs, that they were going to feed on. So they were able to take this waste and produce this really good protein because their larvae is 40% protein, 30% fat. And by feeding them on different products, they can alter this ratio quite amazingly. Now, we're not allowed to feed it on humans yet, but the idea is that we have our own little factories at home, our own little maggot factories, which is so cute. You put in your waste, the flies feast on the waste, the flies have babies, the babies are, they pop into this little chamber, and then you cook them, and you grind them up, and you use them as flour. Now, I've eaten flour. In Germany, you can have pasta made out of insects. You have to be very careful because it is really protein rich, so you have to mix it. But this is amazing. You're giving free protein to the planet. You don't need any water, you don't need anything like that. And what they do if they're feeding on other sorts of waste is that they outcompete the flies that transmit the bad bacteria. So all in all, these flies are amazing. Second story, I love these. I mean, I have to say I am obsessed by maggots, can't help it. I do probably admit that this is a maggot only its mother could love. If you come this close to a maggot, something's gone really wrong in your life. I'm very sorry, because this is one of the most important maggots we use in forensic entomology. So this is great. So we have been looking at forensic entomology for a very long time. 1,000 years ago, plus or minus, there was a magistrate and he was called in to look at a case where there was a dispute and one of the farmers sized another farmer to death, which is a bit harrowing, and he was asked to come in and find out what was going on. Now, he'd already written this fabulous book, The Washing Aways of Wrongs, which I love, and he was beginning to understand how flies worked and what happened. So he goes, okay, everyone bring out your scythes. So they all stood in a row, they brought out the scythes, quite a hot day, and after just 5 to 10 minutes, all the flies went to one side. And the guy was like, how did you know? And so even though he cleaned it, what he thought was really well, he'd left particulate matters of blood. So the flies, just in case you wanted to go down a different career, they can smell a corpse from 9 kilometers away. Just saying. So we have been studying this, and we've now worked out how successful this is. So we can now look at insect succession on corpses. So we can look at how this has developed over time. So we know that they can turn up really quickly, and then as the body changes, as it deteriorates, you have all these different things going at time. So in the tower at the Natural History Museum, we used to keep dead pigs. We're not allowed to do forensic work in the UK on humans, a little bit of grave digging in our past. We're not allowed to go there, but we look at pigs because they're perfect analogues of humans. And this was very useful. We've been studying this, and it came to a head in the UK in 1935 with Dr. Ruxton. Dr. Ruxton, he was a lovely guy. In fact, all his patients still wrote down even after what happened. He was so lovely, he was wonderful. He was married to this lady, and she was very successful in her own right. She did like a bit of high society, though. She did like partying a little bit more than him. And he got jealous, and then one day he snapped and he killed her. Now, being a doctor, he realized that this actually was going to have bad consequences for him. So he chopped up the body, hence the jigsaw killer. Sadly, this was the housekeeper, and she walked in on the act. So she succumbed to the same fate. He, being very clever, he did different things. He chopped up the body, and like all people in England, he drove to Scotland to dispose of the bodies. He dumped them in a place called Moffat. But like all good stories, there were some really bad storms, and the bodies were exposed. And luckily, for us, the first detective on the scene knew of an entomologist. This is in 1935, and he picked the maggots from the corpse and sent them to the entomologist. And the entomologist was able to age these maggots and said they're 12 days old. I know what species they are, I know where they come from, 12 days. And they were able to look at Dr. Ruxton because there was all sorts of other evidence incriminating him, and he drove to Scotland that day. They have the record. So they were like, aha! Piece of advice for anyone giving talks, make sure you clean your fingernails before you have the maggots. These maggots live on my desk. They're the most important maggots in the museum. And they were the birth of forensic entomology in the UK. In the museum, we are doing all sorts of different studies with it. So this is my colleague. They were looking at bodies in suitcases. So they were like, if a suitcase is left out and we've got flies in there, is it because the body came with flies and they bred inside, or have they somehow managed to get the flies across? So they filmed the females, and the females were able to oviposit. She was able to manipulate her ovipositor through the zips. And we bought some of the poshest bags out. They went to Liberty's to buy really nice suitcases, and still these little flies get through. And the maggots are basically plastic. They're like sleeping bags with soup in. They can squeeze through absolutely everything. So they were straight in. And nowadays, we're utilizing this sort of technology. This is a micro-TT scanner. So you remember that life cycle, let's go back to that, and we've got that hidden pupil stage where we don't know what anything's going on. Well, now we do. Now, we can actually see in real time what is going on. So this is the pupa at the beginning, and as it develops, you have this thing called a gas bubble. And we're able to measure the size of the gas bubble, how it develops, and how it then disintegrates, and we can age. So from now on, the police can give you some pupa, you can have a look at it, and you can give them how long it is in hours instead of days or weeks. So it's really hard to get away with it nowadays. But if you want some advice, come and see me afterwards. You can get rid of the body, but I could totally get it. Yeah. Between us, we'd make a fortune. And a third and final story is with Drosophila. Now, most people in this room will have dealt with Drosophila at some point in their life. I will first of all tell you they are not fruit flies. As a taxonomist, this irks me. Not as much as Jurassic Park. That irks me more. Because as somebody who studied flies, they didn't even have a mosquito in the cane. It was a crane fly. And then when they did have a mosquito, several wins on, it was a male, wasn't even blood feeding. And then the species they had was Toxirhynchites, and everybody knows that doesn't even feed on humans. Oh, so really upset me, but I'm moving on from that. I'm okay. But Drosophila is the model organism. If you want to have anything, it's amazing. And Drosophila was really brought into the fore by Thomas Morgan Hunt, and this is his famous lab. This is in, Hunt Morgan. This is his famous lab where about 100 years ago, and I love this. This is a man who wanted to disprove something so much, he proved it. He had read about Gregor Mendel's inherited characteristics, and he was like, nah, it's not going to happen. And he was so adamant, and he did thousands and thousands of replications, and he proved it. Brilliant. And I quite like that. And so he was like, okay, what is it? He's proven that we can have these inherited, these sex-linked characteristics. So suddenly, we've got this mutation that we are able to see by cross-checking and cross-checking and cross-linking that we can actually pass on. Boom, genetics. It was great. And these little flies, we use them for all sorts of things, okay? Why do we like them so much? Well, we just know a lot about them, okay? We've been studying them for over 100 years. There's labs everywhere. I've been to so many Drosophila labs, and it's great because I've asked them, what is Drosophila? And they didn't know it was a fly. So they know everything about these things apart from what they are. They're large but simple. You can tell when you hang around with this fly specialist because they're like, how big was it? It was enormous. Okay, it was that big. Now, they are large. They're not as large as what was originally used, hares. But what was better about these is they liked having sex. Now, that might be quite funny, but it's really useful because they're having sex all the time, and so they're reproducing all the time, and they've got a really short generation, and suddenly billions more flies. So if you're waiting on hares, you've got quite a long gestation period. So you're like, okay. So they've moved on to flies. And in all of this, they're very much like us. Maybe not the latter, but in all the other ways. And we've been looking at these flies now for a very long time. In fact, we're studying them for, we get these flies drunk to understand how alcohol impacts genes. We get these flies, we get them in all sorts of different situations. And my favorite thing nowadays is that we send them into space. I interviewed a lady from NASA, and she's like, nope, that's not how they go into space, Erica. They go in these little fly cassettes. I'll get there in a minute. So flies were the first animals we sent into space, okay? In 1947, they made it to what NASA defines as space, and they have been going up there ever since. There is the fruit fly labs. It annoys me slightly, that. They're up there and they're doing all sorts of research. And one of the big things they're looking at is immunology, and they're looking at the impact on the fly's physiology and how we are going to travel. And one of the really scary things I've read about all of this is that the flies' hearts get weaker, okay? They can't pump as much. So the flies that are bred in space and have come down, they are very weak creatures. So if I ever needed a reason not to go into space, that is one of them, because I quite like my heart, and my heart quite likes this planet. Ooh. And so what I want people to take away is that I've talked about three species of fly, that's it. Okay, we talked about some calophorids, we've talked about some soldier flies, the Stratiomyid, and we've talked about Drosophila. There's 165,000 described. There may be up to another 500,000 species to go. And look at them. You get all these weird variations. I absolutely love this. This is another Stratiomyid, but her bum is wider than she is long. It's great.
In her 2017 work, The Secret Life of Flies, the entomologist Erica McAlister estimated that for each person on Earth, there are how many flies? The figure in question is about the population of Zambia or the Netherlands. You can have 3 million either way.
$6 million?
No, it's 17 million. Amazing, isn't it?
It is, Jeremy. It is. It's very amazing. There's an awful lot of flies out there right now for you to study. So thank you.
Erica, thanks so much for sharing your passion for flies. That was really a fantastic and entertaining talk.
Thank you.
Thank you. Do we have time? We've got time for a few questions. Yeah.
So you didn't really talk about midges.
Oh.
However, I have quite a lot of experience of being a favourite meal for midges. They're not eating chocolate in Scotland. So apart from me, what are they eating and why do you always find them close to water?
Their larvae, a lot of these midges, their larvae are aquatic, so they need that. So you are relatively new in their life cycle, in their life history, so large vertebrates. So if you go to Canada, you feel sorry for the moose. What's really fascinating, I was looking up exsanguination, because everyone looks up exsanguination, don't they? And no human has ever had enough blood removed out of their body by midges or mosquitoes or anything. Mosquitoes in Texas did exsanguinate a cow once. It's cool, isn't it? But for us, we'd need, for mosquitoes, I haven't worked out for midges, but for mosquitoes, we'd need 414,000 feeding us on one blood event. We just don't have that much flesh available. So I've been attacked by my little ladies, but I like to think of me becoming a foster mother. That's the way I look at it.
Okay, anyone else? Yeah.
So apart from bees, what other types or species of flies are beneficial to humans?
All of them. But you're looking at this in the wrong way. What's the point of humans? We look at the benefit. So where do we start? We're beginning to look at, okay, pretty things. I'm not really good at pretty things, but you've seen the metallic colors on blue bottles and things like that. That is structural color, okay? So this is a color that will never fade. It's not like pigment. And we're now developing paints based on structural color. Now this is really cool. Apparently, you can buy a pair of Nike for £80,000. That's not so cool. But what is cool is that they're beginning to paint aeroplanes with this. Now, if you only paint an aeroplane once, because usually it's every year it gets painted, you add paint on, it adds weight. That is bad for the environment. So developing this structural color is really cool. So there's all sorts of different ways they can do it.
Thank you for the brilliant talk, first of all.
Thanks.
What made you want to study flies?
Because I'm nosy. And flies are everywhere. They really get their tarsy, their little feet, into pretty much everything. So when it comes to understanding our planet, We've got to ask the flies. Everything else is just cherries. Flies are basically where it's happening. Sorry, bacteria people in the room. And fungi people, and maybe some of the botanists. The rest of you, nothing.
So speaking of flies and what got you into flies, do you have a favorite fly?
No. That's like asking if you have a favorite child. Obviously, I do. I quite like the venomous ones because people... I'm possibly the worst person ever. Why did you invite me here? I like everything that kills. Or botflies, just because anything that squirts its larvae up the nostrils of a camel. It's arguably, I don't know if I should tell you this name, it's my favorite scientific name on the planet. It's Cephalopina titillator. Exactly. I don't know if Mr. Clark really understood the definition of that word, but they were squirt. I did a gig, to 6,000 people at the Albert Hall talking about squirting botflies up the nostrils. 200 little maggots get squirted up the nostril of a camel. And it does, when the maggots mature and the larvae want to escape and they want to pupate, they will irritate the nasal cavity and then she just snots. And that is one thing I want to see before I die. So maybe that.
Okay, that's really cool.
Thanks.
Thank you very much.
So I'm really disappointed. I came to a Diptera talk and there's no maggot debridement of wounds.
I can't fit it all in. Okay, I love maggot debridement therapy. You can get this on the NHS. It's absolutely fantastic, phenomenal thing. And it's similar flies that are used in forensic. Luckily, these are sterile maggots. There were some really early investigations because a modern debridement therapy came about after the American War of Independence. And they were noticing all these corpses, not corpses, they were alive, all these fallen soldiers that had maggots in their wounds were doing very well. And they were like, what's going on? And the maggots were eating the necrotic flesh. Yay. The guy who first started doing that, so this is all the dead and... If everyone are hungry after this, all this necrotic flesh and the maggots doing well. So they've developed it, and on the NHS you can get tea bags full of maggots, and it's amazing. So they're sterile, but what these maggots do is not only are they getting rid of this necrotic flesh, they're releasing an enzyme that causes your flesh to bind better. This enzyme is getting rid of the bad bacteria. So it's getting rid of MRSA and things like that. So if we go to cases where it's type 2 diabetes and you've got those beautiful, large, ulcerating wounds, and nothing's going to get rid of them, stuff them with maggots. My mum cut her leg open. I was really excited, but apparently the wound wasn't big enough for the maggots. But it's a really good thing, and it's really good because we have so much antibiotic resistance going in. So them and some bacteriophages, we can get together. I know it's all good. See, I can just throw maggots in any situation. Wonderful things.
Thank you. I can go home happy now.
There's one at the top.
All right, we've got a slightly scary question online from... have we got one behind me? No? Yeah. I'll go ahead with the online question.
Oh.
So Jax Slivovich, I think I've pronounced that right, apologies Jax. What health concerns are there regarding the consumption of insects by humans, and is there a possibility of a genetic mutation causing a pandemic?
Wow, it's a lot of responsibility in one question. They're very different to us. So the nice thing is a lot of insect diseases, we can't pick up those diseases because they are very different. So there's less. But the reason we're not letting it go ahead is that we are being very, very safe. So we can eat them fine if it's on decomposing plant material. We're a little bit more concerned when they're on decomposing mammals, because then they could be transferring something that we don't know about. So we're being very, very safe with that. But dead vegetables, eat them, and then we can eat the maggots. There's probably a more technical way of replying to that.
Okay, one more question at the back.
Yeah, it was off the back of the question down here about the maggots. What benefit is there to the maggots in producing an enzyme that would heal the skin?
I don't think they thought it through. I don't think evolution works like that. It just happened. I think it's just one of those things. It's a happy benefit.
Yeah, okay.
Yeah.
Okay, thanks so much. Brilliant questions. Can we thank Erica again for a fantastic talk? Thank you so much. Okay, folks, so we're right at the end of time. So we've got a few thank yous to make. I think we'll combine them all into one mass round of applause. First of all, to all of you guys here on site and also that have been tuning in on Zoom, it's your enthusiasm that really contributes to the success of this event. So thanks so much, really appreciated. Also want to thank all the speakers today. They've done such a tremendous job, putting so much hard work to come and talk to us today. So thanks a lot. And then finally, the organization of this event started about a year ago today. So I'd like to say a big thank you to Sam and his committee team for organizing such a fantastic event that grows year upon year. So thanks again, guys. See you next year.